Hello, everyone. My name is Martin. I'm from Lausanne, and this below is Uwe from Dresden. And we're today giving the tutorial on deep learning-based object segmentation and image restoration with StarDist and CSBDeep. And the rough outline can be found in the GitHub repo that you hopefully all have already found out the link to. This should include all the notebooks and everything that we will speak about in terms of resources. And there's again a little tentative schedule. So we're now in the introduction, short introduction. Then, as I already mentioned in the email, there will be a slot where each of you is presenting their images for 1 minute and 1 slide. And I will just ask the names in succession and you just share your screen. I hope this is fun with everyone. Then I'll give a little bit of an overview talk of image restoration and object detection with CSBDeep and StarDist. Then Uwe will speak a little bit more about advanced topics. Then there will be a break after 2 hours. So yeah, in 1 hour and 50 minutes-ish for 15 minutes. And then we're gonna assign people to breakout rooms and work on actually notebook tutorials that we prepared for you. And this is kind of a little bit depending on your use case, and there will be different groups dedicated to different use cases that you might have. And I hope that is interesting for you to get a little bit of dive into how you could use that in practice. And then in the end, we'll do a little wrap-up session where you can answer more questions. And as again, if there are any questions during the talk, feel free to just interrupt me or Uwe and make it as interactive as possible. So just to give you a short overview of what kind of data we're looking at. I mean, we just looked at for 40 minutes on the most beautiful data already. And this is just a short overview slide of what is the phenomenon of different images that are happening in 2D and 3D microscopy, but not only light microscopy as well, electron microscopy. And these are typically the data that you see and have certain issues with. And we're going to talk about two approaches for two certain subclass of problems that you might have when looking at this data. And these are object detection and image restoration. And from a very high level point of view, you can think of object detection or segmentation as your input will always be an image, and an image is a almost continuous valued 2D or 3D array of pixel intensity values, right? So every pixel would have almost continuous value of the pixel intensity at this point. And for object segmentation or cell detection, what you want is the input is a continuous valued image and the output will be a discrete valued image, right? So every pixel will be assigned one of many discrete classes. And this could be, for instance, foreground or background, but this could be like different pixel IDs or different cell IDs, right? But it's always like a discrete set of cells. And if that's the case, people speak about segmentation. Well, what? That's I have to move your faces away, but I can't see my mouse. Okay, now. If now the case is that you have, again, like a continuous valued input image, but you want to denoise it or you want to transform it into another image, which is still containing pixel intensity. So your input is again a continuous valued image and the output is again a continuous valued image of pixel intensities. For instance, if you want to denoise or deconvolve an image, then this is called regression because you're not classifying pixels in individual discrete classes, but you're really regressing on the values of pixel intensities. And it is well called image to image, or in a broader aspect, the problem that you saw there is image restoration, right? So these are two conceptually a little bit different subtasks. So once you want to predict for each pixel a discrete set of classes, and the other one you want to predict actually intensities, or you want to predict the continuous valued function, which is regression. And we're going to speak about both of them. And the first one will be solved with the first one will be object detection with StarDist, and the second one will be CSBDeep or KER, content-aware image restoration. So conceptually a little bit different. So in the first one, we speak about this image restoration aspect that we're going to solve with, or going to try to tackle with deep learning-based approach. And here again, just a reminder: everybody of you does microscopy, so you know about the trade-offs and specifically in live cell imaging, right? That you cannot have you have a certain photon budget, right? And you cannot image with at any dimension at the highest level of accuracy and quality. So you always have to haggle light exposure speed and spatial resolution, which induces some loss of image quality that you have to cope with, right? Just to ensure that your sample is healthy after the imaging. And this pertains to the light dosage that you can use, right? So instead of crisp, high valued, high intensity image, you only get a very noisy copy of that or noisy versions of that. But it as well applies to the speed with which you can image that typically limits the number of Z-planes in 3D lifestyle imaging, for instance, and thereby a loss of axial information, and as well the optical properties of your microscope itself, which always induces a limited optical resolution that you have to get around with deconvolution or with other image resolution enhancement methods. And just to give you a short example of that, so this was flatform imaging that we did back in Dresden, and the flatform is very extremely light sensitive. So if you, and this is an anesthetized worm, so if you put a light with which you can see every cell individually in this imagery, the worm will just twitch and shake so much that you can do no tracking, no segmentation, and every image analysis on that is practically impossible. So you had to use way less laser power than that just to make the worm somehow happy. And if you do that, you just, of course, end up with lower camera exposure and lower laser power. And then the worm becomes quite resty, but on the other hand, the images become quite noisy, right? So, and it's very hard to just with anything classically to image, to segment those cells or to analyze those cells more, right? And the question is, can you, with that light condition, can you kind of recover the original clean ground truth signal without shining too much light on this one, right? And that's an approach which we took with what we termed then content-aware image restoration. And the idea is quite simple. You just go to the microscope and you generate and collect pairs of low and high SNR quality images of the same structure. So we went to the microscope and fixed some worms and imaged the same part of the worm just with different microscopy settings, and one which was with low camera exposure and low laser light, and the other with very high laser power and very long camera exposure that you could not use during live cell imaging, but you can, of course, if the worm is fixed. And then you just train a regression model, in this case, a convolutional neural network, to predict, given low quality, low SNR input images, predict the high quality output. And then you validate this regression model that you got. So in this case, a residual UNet, you validate that on stacks from worms that you haven't used during training and see how well the results actually match up with the high SNR images that you could acquire on fixed worms. And this is just how it looks like. So we imaged 3 different low conditions and 1 high condition, assembled stacks that are corresponding. So these are showing exactly the same nuclei at the same position of the microscope, just with different imaging conditions. And then you train a network. And if you do that, well, if you do that, you can then put it on live cell imaging of new worms and just denoise them quite satisfactory. So you can then make out individual cells, and all this noisy background that typically hampers any normal thresholding on what you would do with the image. It's just gone. This, of course, works in 3D, so you can get rid of most of the clutter in a tiled, in this case, tiled volumetric acquisition of the worm. And this is just to show you that you actually recover all of the nuclei and are not painting nuclei in there where there should be none. So if you zoom in, you see one of them is the input, one of them is the restoration, and the third is the actually ground truth of a fixed worm.
Martin?
Yes.
So if you're doing that for a given pixel, its value is going to be either increased or decreased. Doing that, could you use that for segmentation in one step?
Exactly. If you want to if your final goal is segmenting with something extremely powerful and where you have annotations already, you could do that in one step, right? A segmentation network, essentially regressing on the pixel values, it's a way much harder task than just to find out is it inside or outside of a nuclei, right? So this is an easier task because the network could just learn a content-aware threshold. And in some sense, if you only want to do this, it's way better to just directly segment noisy data. So this will always work better than if you regress on the actual pixel values, right? So if that's all you need, I would just right away segment. Yes.
Sorry, maybe if I can jump in here. Did you have an extra correction for the, let's say, non-homogeneous field? because I see that after restoration, it also gone. There was an extra step, or the network also took care of that?
No, this was because that was actually exactly. So the stripes you see, this is from the vignetting of the tiling, right? And this only appeared in the low SNR. In the high SNR, this vignetting was not present. So it kind of learned that whenever there's a vignetting in the low, it should restore the high according to what it has seen in the ground truth, right? So this is an interesting question. So this was done as well.
It's just strange for me, like, how was it that, because I guess you had to, like, when you train on small patches of this image, and it still managed to learn the vignetting somehow.
Yes, because the patches were, so some of the patches had actually the boundary region, because we made sure that we sampled, like we always typically randomly sample from a given larger stack. And some of those patches had just the boundary with it, with the vignetting. So this was included in the training data.
Yeah, but if you can also adjust for that. You make it easier for the network to learn.
Sure.
So if you have some flat
fielding,
just do it. Yeah.
If you have a flat field anyhow, which you probably have, if you have a strong vignetting, then doing that before is of course always better, right?
Yeah, and for the earlier question, right, this image restoration is useful if you can then use a traditional segmentation pipeline, for example, a non-learned pipeline, or if you just, of course, have the images to look at by a human, it's much easier, right? So it depends. If you want to do segmentation ultimately and you can annotate your data or have segmentation labels, you can do it directly. But also sometimes you can use this image restoration to even be able to annotate. Sometimes you cannot even see the nuclei clearly in the low SNR. So you could use the restoration approach to then help you annotate and then use that in the second step and to directly train a segmentation network on the low SNR data.
Gotcha. So this is essentially what Mark was saying. If you look at this, right, annotating those nuclei is super hard. If you want to segment those cells, just annotation of the low SNR is super hard. If you denoise it before and then just do a simple thresholding, it would already get you a lot of those masks for free, which you then can curate way easier than it would be possible in the original one. And in this way, you can generate quite easily like the annotation mask for a segmentation step. That is what you're actually after at the end, right? So that's what Uwe just described. Okay, of course, like so far you always needed like, yes.
Yeah, I had a quick question. I guess I missed it. So for the network, what do you provide? Do you have to provide like annotate ground truth? Or just like a high resolution ground truth, or just one start from the noise one?
So far you always need it, and it's not pertaining only to denoising, but it works as well. And I will just quickly show you another example. It is well for upsampling or for deconvolution, right? You always have to provide like these pairs of the same scene, just taking under different conditions. And it could be low SNR, high SNR, or low SNR downsampled and high SNR upsampled. But you always have to have these two pairs of the same scene, right? Which is of course a limitation because sometimes you don't have that, right? And this slide is just showing you what happens if you don't have that. So. Okay. Was this the answer?
Yeah, because I was wondering, what if we don't have that upsampling images? What's going to happen?
Well, if you don't have the upsampled image, so if you really want to do resolution, or if you want to improve the resolution, then it's hard to do that without ground truth. But in the case of denoising, you actually can get away, or you can get. There are methods that are actually that I want to speak about, or that I will speak about now, that can get away this strong constraint that you need ground truth. Right?
I mean, the method you're describing, where you're doing a high resolution of a fixed sample, comparing it to a live sample, is, to my mind, a bit dangerous. Because that fixing will create all kinds of artifacts, both in shape and in fluorescence distributions.
So for the fluorescence distribution, I would agree, because like every fluorophore is kind of has its own characteristic, right? And this all is not meant to, I mean, you should not photon count with it anyhow, because it's essentially like any network will do a nonlinear transformation of your signal in some way. It has to do that. So if you really want to photon count and do some correlation measurements and really want to count what is the number of photons coming from the sample, of course, just due to the normalization, this wouldn't work, right? Because that's in the raw data, right? So you should go back, either restore it, do some segmentation, and then go back to the original data and measure what is the region of interest that you're interested in, and then count the photons in there, right?
Yes.
And of course, and I will speak about the validation aspect. You always have to make sure that whenever you train your network on, the data is similar to the data you then apply it on, right? Because that is the core assumption for almost everything in machine learning, and particularly that's the core assumption in image restoration, right? Because if you don't do that, like weird things will happen, and this should not. That you try to avoid that. But I will speak about that later.
Yeah, it was more about the philosophical, or philosophical is a big word, whether you should add noise to an image and try to, I don't know, model the noise you get and then add more of it, or is fixing and then having a good resolution image, what is the better strategy?
So far so my guesstimate would be that you're never as good in modeling noise as an actual camera, because most noise models are quite simplistic. They do some shot noise, some Poisson noise, and some Gaussian background that might be pixel depending, but any sensor is a little bit more so a CMOS sensor of a camera is a little bit more complicated than this, and it's hard to capture that all in a perfect well model. So I would just, so typically if you add synthetic noise to images, like it's super easy for a network to learn what is Gaussian noise and what not. And this is not always present in real noise, right? And real noise is not always perfectly Gaussian, and it's not always perfectly a shot noise or Poissonian noise. And that's, if you make it too simple, or if your noise model is too simple, then a network will be just able to learn that and look at textures and characteristics and correlations of the specific noise model that you use, that you as a human would not be able to draw any information from, but a network would. So I would always stay away from that. And it's not necessarily that you have to do fixed ones. So this was just, you don't have to do fixed versus live, but of course you can just do the same in live cell imaging. And I show you an example afterwards. You can just do for some time steps at the beginning where you don't care about phototoxicity. You just sacrifice some of those, or like you don't do long time lapse imaging, but just do a short time lapse where you interleave those conditions and just make that in a quick succession, right? So you can use the same live cell reporter or the same fluorophore as you would use during the actual imaging. And that works out quite nice, or this should work way better than just adding noise. I mean, if you can acquire clean ground truth anyhow, then it's relatively cheap to just acquire a super fast camera exposed image before, right?
Okay. Yeah, I think the fixation was a special thing for the worm who twitched so much that otherwise it wasn't possible. But in general, if you can do it all with live imaging, you should do that. And so it's the same conditions, right? And sometimes if it's moving too fast, then Martin's going to speak about this noise to noise now, where even if you have two paired low SNR images, you can still work with that for denoising. So in your live cell imaging situation, it may not be possible to use like a long exposure because the sample moves too quickly, but even then you can do this. And I think, yeah, Martin's going to talk about that now.
Okay, so if you don't have this ground truth, right? So in most cases, you actually don't have that because you want to denoise images you already acquired, right? Or like your sample just doesn't allow to acquire like paired images. There are actually two interesting approaches that popped up in the last 2 years that get away this constraint, right? The first is called noise to noise, and that is using two noisy images of the same scene. So you have the same object, the same sample, and you acquire quick images that are very noisy with very low exposure, for instance, quickly in succession, such that they show the same scene, but they have a different noise each time, right? And the interesting part is if you just train the same thing, the same approach as before, just using the noisy second image as the output, which is actually not a clean output or a high SNR output, but it's just another noisy instance of the same scene. But you use the same method and train a network to try to recover the second noisy image from the first with different noise instances. What the network actually can do, it's super hard, it's actually impossible to predict pixel-wise independent noise from different instances. So it cannot really recreate the noise patterns, that it has no information how to do that. The only thing it really can recover is the underlying clean signal or the mean of that noise distribution, right? And that's what it does actually. So it's very interesting that it actually works in this way. But so you provide just two noisy inputs. One you use as the input, the other you use as the output, train a network to recover one from the other. And what the network has to do, or the best thing it can do, is just clean up the image and provide like an average intensity, averaged or the mean intensity according to your noise model. And this is your ground truth, right? And here you see like the third image of the koala is like what can be recovered from the two input images, which is quite astonishing. And of course, sometimes you don't even have two noisy inputs, or you don't even have two noisy copies of your same scene, because you just can acquire a single image, or you did that already. And that's like a situation that is quite common and is tackled by two methods called noise to void and noise to self from Florian's lab and Le Croyer's lab. And what they do is they, just as an input, just accept a single noisy image, but they block out a single for each pixel you get as an input, or each pixel sees everything apart itself and has to restore its value. And this way, the network is forced to, just given the neighborhood of the pixel, to infer a meaningful estimate for the center's pixel intensity. And that is typically the estimate of that. The best estimate it can do is, again, the mean of this pixel value. And this mean of this pixel value is, again, the clean ground truth. And this works quite well. And there's a Fiji plugin. And if that's your situation, you don't have any paired images of that, I would highly recommend using that for your data. And as I said, the advantages are that there is no clean ground truth necessary, and you can predict and train on the same images. And the only drawback so far is that so far it only works really well for denoising, and it assumes pixel independent noise, right? So if you have for some detectors like spinning disks or SCMOS, there's the noise is correlated along certain substructures of your sensor. So if you know, maybe if you do imaging with SCMOS, you see that the pixel gain and noise is actually correlated along stripes in your camera, and that's something that you probably will see then popping up in the recovered image, because it's essentially it's so to speak part of your image that you want to recover, because it's not pixel independent noise anymore. But suddenly you have correlations that go farther than 2 pixels. And just because I mentioned that this all doesn't only apply to denoising, this is an example from other people in Dresden, from collaborators in Dresden that wanted to image like the epithelial layer of flywing time lapses in the imaginal disk. And that's the stack quality they could get according to their needs for sample health. So this is a XY slice at the left, and an XZ slice, an axial slice on the right. And they were interested in the shape and outline of this cell sheet, of this membrane layer at top, and the folding and the biophysics of this folding. And they, of course, wanted to do the imaging for quite some time. So they imaged several hours of that with a very high succession of stacks time-wise. And of course, then they were limited in the SNR that they could achieve and the actual sampling. So they had to really do very, very coarse Z steps with very low camera exposure. And that were the stacks they're getting. And they were not sufficient to really trace out every of those membranes. But of course, you can do the same trick by just using some of those stacks at the beginning, quite fast in succession, acquire low undersampled or less well sampled in Z, and a high sampled in Z and very high camera exposure stack directly afterwards, and combine those stacks, gathering paired images, and then trying to recover one from the other. And that's what resulted in stacks that are actually now, with classical methods that they already had in place in their workflows, were able to manage to then segment those cells quite accurately, and then trace like clones when you go over time quite successfully, which could not be done in the original data. That's just an example for a task that is not only denoising, but as well upsampling. Of course, like we already mentioned that, like there's always, whenever you do machine learning and supervised machine learning in particular, so this paired images, there's always this core assumption that your training data distribution, so the one that you assemble for training a model, equals or is similar to your prediction data set. So the stacks that you then later on predict. And that has to be, in this case for microscopy, that means that you have to have the same organism, the same structures. It has to have similar SNR, which the stacks you want to apply them on, and then the one that you train for, similar resolution, similar microscope settings. So essentially for every experiment, for every experimental line that you do, so for instance, if you want to do denoising of membranes, you always have to use a dedicated network or a dedicated restoration pipeline that is specific to this experimental condition that you use. So these are caveats, and typically how you check for that is, and this is very common applies to almost any machine learning based pipeline, but in this case for image restoration in particular, but again for StarDist too, to make that, to ensure that on the trainings or during model training or during actually your pipeline that you're building, you always split your whole data set that consists of paired images of input and output. You always split that into what is called a training set and something what is called a test set. And only the training set is actually used during the model optimization. So this is constantly accessed during training. And at the end, after everything is done, your model is finished, and you then put it onto your Fiji plugin and apply it to new stacks. Then you put it on, or you evaluate that on a test set, which are stacks that you haven't seen during training, that you didn't use for your model optimization, and that were not even from the same sample, right? Which is very important. And this gives you kind of an unbiased check on whether you're actually succeeding in restoring it correctly, or whether there are problems, artifacts popping up, or so forth. And there's a little detail, an additional detail, that even the normal training set, you even split then again in a proper training set and a validation set. And the validation set is actually used during model training to evaluate the model. It's not used during training, but each epoch, so each like subset of runs of the model, it constantly checks its performance on a holdout validation set, which is a subset of the training set. And it's only used to check when should you stop training, when is your training enough, when do you actually overfit. So overfit means that your network is actually, it's too good, it's getting too good on the training set, and it's losing performance on the test set. So it's concentrating very heavily on your training set, the intricacies and textures of your training set, but it's not focusing actually on the things that would generalize to unseen data. And you want to always avoid overfitting, and the validation set is there to check for that. And we will see later in the notebooks that this is like a common thing, a common split to do at the beginning. So you always want to split your data set into these three subsets. Okay, so if you're interested in that, there's this webpage that you can visit. And if there are any questions, typically the best place is the forum. And if you use the tag care, we will see it right away and hopefully answer. And there are just two projects that are worth, that I urge you to check out. So one is Zero Cost Deep Learning for Microscopy from the Henriques group. So they have some collab notebooks that implement a lot of deep learning based, not only restoration as well as segmentation pipelines bundled in a collab notebook infrastructure. And then there's Noise to Void, which does that denoising step without clean ground truth, which is, I think, very cool. Okay, now we come to object segmentation. Are there any questions so far? If not, then I will just directly jump into that part. Okay, now instead of regressing on pixel intensities, we want to tackle a different problem that is quite prevalent, right? Which is image segmentation or nuclei segmentation in particular. And here the problem is that you're given like an input image of potentially very crowded cells that are touching and a lot of noise, a lot of out of focus blur. And there are essentially two main sub-problems of segmentation that you could think of. The first is what is called dense segmentation. And dense segmentation means you're just interested in categorizing each pixel into a small number of classes. And the most simple is just two classes, foreground, background. So you would just ask for what is nuclei, or what are cells in general, and what is background. So this would result in a binary image. But what is a little bit more of a complicated task is what most people actually want. They don't only want to see what is foreground, background, or want to know what is actually just a background class and what is foreground, but they want to assign each individual pixel to a different cell instance, right? So you want to separate those cells. And this is called instance segmentation because you want to segment each instance of an object, right? Not only just lump class of being object or not, but even different instances should be separated, right? And this is what we typically want when we ask about or feeling ourselves confronted with those images, and because then we want to correlate on a per cell level individual expression or other markers, right? And there are several common deep learning based approaches for this instance segmentation in particular, in general, and nuclei segmentation in particular. And they can be roughly categorized into two groups. And the first one is called the bottom-up approach. And this is like you first do this dense segmentation. So you first do kind of a foreground background segmentation or another class for the object boundary. And then you try to, from this foreground background or dense segmentation, you try to then discern individual objects with watershed, with like getting seeds via distance map or and then doing a watershed on that. So for instance, like a lot of people use UNets for that, or elastic random forest classifiers that give you like a foreground background mask and then try to separate it with a watershed or with some other seeded methods. And then there is the top-down approach. And in the top-down approach, you first try to localize your objects. So you first try to disentanglement by localizing your objects. And this is typically done with bounding boxes. And then you try to refine this bounding box to a shape that is more appropriate to your nuclei, right? And one method that is quite popular in the computer vision field, so for natural photographs of people and bicycles and so forth, it's called mask RSNN, and that's one of those top-down approaches. There are other methods popping up in recent years, so affinity-based methods from the Heidelberg people and in Berlin, and embedding-based methods. For instance, CellPose that does it a little bit different by following flows or following flows, inward-looking flows. But these two things are mainly what are prevalent approaches to segmentation in deep learning. And of course, they have, as I already mentioned, some problems in the case that we're actually interested in of very crowded cells. So the first problem with the bottom-up approach is that although the segmentation is fine, this disentanglement step is sometimes failing, especially for those cases, right? You find, instead of 3 seeds in this case, you only find 1. And then, of course, this results in cells being lumped together that you don't want to be lumped together, right? And in the top-down approach, so if you want to localize cells with a bounding box, of course, you have similar problems because bounding boxes are not really tight for each cell. So you have overlapping bounding boxes for cells that are different instances, but they overlap so much that this detector would think it's just a single cell, right? So it would be categorized again as a single cell, which is not good. Of course, what you can do, you can try to merge those two approaches in a way that you still have kind of a localization step, but not with bounding boxes, but with something that is more appropriate for a cell shape. And that's essentially what StarDist is. And StarDist predicts, instead of bounding boxes, it predicts 2 things. It first predicts like object center probabilities. And this is just like a 0 to 1 probability map for each pixel to be the center of an object. So it peaks at every center of the ground truth that you provide. So this is the annotation. So it tries to predict where's the center of a cell. And the other thing it does, it tries to predict radial distances. So for each pixel, you predict distances towards the boundary, and you do that for a certain set of predefined directions. In this case, for instance, 16, but typically this is like 32 distances. So you get 32 values per pixel describing exactly the shape of the nuclei or the cell at this position. And thereby you have both actually of the bottom up and the top down in one single step. And you do the disentanglement and the precise outlining in one single step. So that's essentially the approach. And you can do that end to end. So the input, again, is a normal image. It can be 2D or 3D. And the output is the object probabilities and the distance maps in one big block at the end per pixel, right? So per pixel, you get 1 value for the object probability and 32 values or 48 or 16 for the distances, right? So you get a whole column output per pixel, right? Of course, you have then suddenly, in your complete images, you have 256 by 256 times 32 distances that not all correspond to different objects, right? So you have to find a way to kind of disentangle those overlapping objects that you get. And this is a step called non-maximum suppression, NMS. And it just actually looks for the most probable center of an object, draws the star convex polygon around it, and then looks for all the other polygons that are similar, belonging to the same cell and overlapping with a certain threshold, and just discards them. So it kind of, and this is depicted here. So you have initially you have a lot of potentially conflicting objects, but you keep the one with the highest probability and remove all those that are overlapping above a certain threshold, right? Which makes sense because they probably belong to the same cell. But I just mentioned that you know that this step is called non-maximum suppression, and it's in the whole StarDist pipeline, roughly like 50% of the whole effort and time spent is spent on the NMS. And the first step is the actually GPU accelerated prediction, and the second step is the NMS that tries to get around those conflicting polygon hypotheses.
Martin?
Of course, there's only yes.
Coming back to that overlapping data set, is it important to have that in the training? Do you have to annotate overlapping cells? Is that super important, or can you just do the easy bit and okay?
So exactly. You don't because you typically even annotate with a label mask, and then it's even not really possible to do that in a single image represented as a single image, like overlapping objects. So you don't typically do that. Actually, in principle, you could do it with StarDist, because StarDist can predict polygons that are overlapping. But the initial representation of that, your labeling, your annotations that you do, is typically done non-overlapping, right? And it will figure that out, that actually two things should maybe overlap a bit, but then that's not something that you need to do. No, it's hard to annotate. It's already hard to annotate these kind of images, but with overlap, that would be, I think, a bit too much. Yeah, so you don't annotate with overlap, yeah, typically.
Okay, but so you still have to annotate the hard cases if you want to get them segmented well, right? So there's no way around it.
Like this.
But the nice thing is, even if you annotate, all your annotations will be non-overlapping, but currently, because it's not technically implemented otherwise. But still, during prediction, there could be multiple hypotheses that are overlapping, and you can then choose to get those overlapping results if you want them. Okay.
Of course, your object of interest that you want to annotate has to be able to be represented with such an approach. And these objects are called star convex. And these are not necessarily, it's a little bit a weaker constraint than being convex, but it kind of just means that you can find in whatever you want to be represented by that approach, you can find a center, one point in the middle at least, where you can see all the boundary along this predefined rays, right? And this includes those green shapes, so it's not necessarily convex, so it can have bumps that are outlined like I showed it, but it cannot be shapes that are looking like the orange ones, right? Where you cannot see the boundary from, when you cannot find a single point where you can see all the boundary, like these protrusion shapes, right? These are not star convex, and of course they don't necessarily fall into the realm of the StarDist approach, because you cannot represent them with this radial distances that you put on the center point, right? And these are the prediction steps. So you get an input, you predict the object probabilities and the distance map, as I mentioned before. Then you get a lot of an overcomplete set of polygon candidates, which you then filter out in this non-maximum suppression step, right? And get the final result. And this is roughly the same in 2D or 3D. This is just some example of lymphoma cells in a dish where you see it's very, very, very crowded and a lot of out of focus. And I think there 4 to 6 crops were annotated of 256 pixels each, and it gave a pretty good segmentation, although it was quite challenging in this case. And it works on the whole image, and I will briefly mention that later. The same applies for histopathology. So this was a pre-trained network that we did on some totally different other data set, and here again you can do that on the whole slide and segment the nuclei quite with sufficient accuracy at the whole slide level, right? You get like 350,000 nuclei and 6 million candidates that needed to be filtered. The same approach, so again, basically. Yes,
Martin. Sorry, again coming back to the annotation. Sure. and the thing. So in general, when you're doing a classification or something like that, you try to have as many test cases in your different classes, so that you don't overrepresent one class over the other. Here, for instance, when you were training on these nuclei, you have some which are out of focus. Where I mean, you must do some kind of QC when you're annotating. What would you recommend? Can you have general recommendations, and whether you should make a training set which is largely skewed to the very clear, where you're very sure? Should you try to avoid the things where you're not very sure, so to not confuse the model?
That is a super interesting question, because it's again a little bit more philosophical, right? I mean, typically you want to annotate those that you at the end want to be represented and found out, right? But of course, there are cases that you would love to be found out without you being sure. And that is almost something like for a nuclei which is super diffuse in the background, where you as a human annotator wouldn't be sure, is this a nuclei or not? And the network or any segmentation pipeline would give you a result that says it's a nuclei. Would you be satisfied with that or not? Because you would have to always compare it to your own annotations, right? Yeah,
I mean, philosophically speaking, what I want to do is to have, so, you know, most people will believe in their own opinion, at least, or else it's not an opinion, right? The problem is with people is that the opinion changes all the time. I know mine does. So today that is a nucleus, tomorrow is not a nucleus. And what I want to do is to ensure that my model makes always the same mistake, so I don't then care. Because I just have a certain percentage of error, and that will propagate all the time through all the conditions, and I don't have differences which are due to my mood.
That's a good question. I mean, it kind of pertains to the question of inter-rater variability anyhow. So if you give the same, so if you give, for instance, this crop to 3 different people and just ask to annotate them, what is even the differences between those 3 annotations, right? And I guess in this case, it would be already like 10% of the cell masks would probably be some say some is a cell in the background, as you mentioned, right? Even yourself doing 2 or 3, if you would do it once again for a different image, for the same image, probably the result of your annotations would be different each time, right? And that is, and I mean, a network or a deep learning based pipeline would kind of just trying to recapitulate that. And of course, we'll give you always the same result, but this result is of course biased towards your annotation selection, right? But it's not something that you can kind of factor out. It's super hard in general.
Well, can you, I mean, that's again, the interoperator differences, can you get more robust models if a lot of different people annotate the data so that the error averages out, and then you hope that the median is going to be the best goal?
That is something that is super interesting, and it's not, I mean, we haven't implemented such an approach, but it's certainly interesting to, for instance, think, okay, you have 3 annotators, and the actually ground truth is always like the consensus vote, and you want to signify cells that are like wherever cells annotations are differing, that's probably the hard cases and not the ambiguous cases. And you probably want to highlight those cases in the final result too, because that would be probably nice to have. But currently we haven't done that. So
yeah, I think comments about that. So there are methods that try to fuse several annotations. So these things exist, but typically people don't even have the time to do 1 annotation of the data, let alone several, because it's just too much work. But if you really cared about that, you could do that. And the other thing is we're currently implementing some sort of additional classification in contrast to just finding all the objects. So you could, in principle, the things where you're not sure, you could give them, you could annotate them as a different class, like unsure nucleus or something. And then later the network could give you a class probability. Is this like a nice nucleus or a not so nice one, right? And then you could then use a threshold based on how sure the network thinks this is a nice nucleus or something. It's just an idea. We haven't tried that, but Martin will mention that we're currently working on some extension that also it gives you a class for each nucleus in addition to just finding it.
So you could label a different, like hard cases, you could just label with a different class, so to speak, right?
In order to filter them out later if you, because, right, I mean, if you never want the out of focus one, just don't annotate them. And then this object probability thing will take care of that and give it a low one, and it will not be picked up later. But if you want them sometimes, you could then also give it a different class, and then you can filter them out on demand based on the application. So these are options, right? I mean, sometimes you just need to try it out and see what happens. It's very hard to give like general guidelines.
Okay. So the same approach basically works in 3D too. The only additional considerations that you have to do, instead of now suddenly like 32 rays that are in 2D, you have to give a lot more rays to represent a shape in 3D. So we typically use 96 that are kind of distributed in a golden spiral, Fibonacci kind type of lattice on the sphere. So they're equally distributed on the sphere. And additionally, you have to take care of data anisotropy. So typically your Z resolution or the pixel size in Z is like 4 to 6 times higher than it is in XY. So everything, if you just look at your raw stacks without actually proper reslicing them, even a single sphere would look like a prolate object, right? And that has to be kind of factored in, but that's automatically done, at least in the notebooks and in the models that we're gonna use. But you have to make sure that this is this is something that takes place. And the other one is the non-maximum suppression, because suddenly now you have not only to suppress, you have to filter out the candidates that are 2D polygons, but now you have 3D polyhedra. So it's a bit more computational involved to do that, but that's not something that you should take care. We try to solve that. Yes.
Yes, I have a question about calculating the distances. So you calculate them between like the pixel that you are looking at and what?
So actually you go for every, you have your given input and the segmentation ground truth, right? And you now go to every pixel, for every pixel in the ground truth, right? You look at each of those different rays that you have or directions that you have. You go along this ray and record the distance and which time like your boundary or your segmentation mask class changes, right? So if you're inside of a ground truth object, you look along this ray until you're not this class anymore. And this is the distance to the border, right? And you do that for every pixel, for every direction. Right?
Okay, so I didn't catch that as after the segmentation. So once we already know what is like
this is done automatically, right? So you annotate only just the foreground background. So this is all done under the hood, given your
because you don't really know what's the actual center of the object. You just have the probability. So do you just go to the one with the highest probability, or?
So it's calculated for every pixel, because even if you're close, if you're not in the center, if you're close to a membrane, or if you're close to a border of an object, towards that border or close to the border in this direction, you have a pretty good notion of what is the distance, but not on the other hand, right? So even for those pixels, you can calculate this distance, right? But they're just typically not used if the center probability prevails, right? But you do that still for every pixel, and you get a center probability for every pixel, and you just, at the end, you just choose for every of those pixels what is the most likely and only use the distances of those. But all of them get predicted. And they kind of then get stitched together. So you have kind of that's why you have so many polygon candidates, because essentially every pixel that is inside of or that is roughly inside of an object will vote for one of those polygon or polyhedra, right? You get a lot of candidates.
Yeah, thank you. Yeah, I think that's a common source of confusion, because in these diagrams, you typically see only the center, the rays, going out of the center of an object, but really every pixel is voting for a polygon that describes the object that it belongs to. So there are lots of them. And these object probabilities are just the scores, right? And the ones with the higher score have precedence over the ones with lower scores. But that's all. It's just a weight that gets assigned, and that's used in this non-maximum suppression to see which of those polygon predictions will be favored.
So this is, for instance, one of the distance maps that will get constructed during ground truth. So this is from this ground truth object, you calculate this, and this is for every pixel. So every value here indicates what is the distance of that pixel towards the right direction to the next boundary, right? And you see everything that is on this side is very high. So this has a high distance along this because it's farthest away for each ground truth object. And at everything that is close to this boundary along this predefined set to the right direction is a low value, right? And you assemble, and this is a dense image, and you have for every direction, you have one of those images.
And in your case, the distribution of the intensities in the object are almost uniform. So in my case, I have only membrane markers. Can it be like the case and StarDist cannot work properly, or with proper training, I can get some good results as well?
If you only have membrane, or instead of nuclei marker?
Yeah, I have membrane markers only, so I have only borders, and inside it's almost the same as background.
But this doesn't matter, right? As long as, so essentially it doesn't care if you're inside of a solid object or if you're inside of a hole that only sees the membrane. As long as your field of view, and we will come to that later, as long as the network sees the whole object, it knows, like if the center pixel of something that is a membrane is able to, the network at this position is able to see the border, it can infer the distance to this border, right? And that's the only thing that matters. So it will work. So I think, some colleague of you tried it. I think Mangal tried it on Flywing, right? And it works on membrane marker of a Flywing equally well, right?
Okay.
Yeah, of course, your label annotation must be the inside, right? That's the important thing, right? You need to label it as if you were labeling the inside of your cell. But yeah, but if it's possible to predict this, or as a human, you can do it, right? So there's a chance a neural network can do it as well from the raw image. So in some sense, it really doesn't matter what the raw image is. As long as you as a human can do it from the raw image, the annotation, then there's a chance the neural network can learn it as well.
And so would you say that if you annotate such an image as Anna was showing, you go all the way to the plasma membrane marker without including the plasma membrane marker, so that you then by subtraction get the plasma membrane?
You could do it like this. I mean, this depends what you want to do at the end, right? If you want to include the plasma membrane marker in your final segmentation or not. But I think it's equally easy for the network to learn, right? To include the membrane, like this 2-3 pixels that are consisting of the membrane or not.
Yeah.
So both, you can do both, right? It depends what you annotate.
Be consistent in your annotation then, but still,
exactly.
If it's really 1, 2, 3 pixel difference between including or not including membrane, then maybe you shouldn't maybe also rely on this being super accurate, especially if you, like, we always have these rounded shapes that get predicted, right? So the shape fidelity on the borders is not super high based on, so you can use more arrays, but in general, I would not rely on the missing space between two nuclei that are predicted and to be super accurate.
I mean, there are ways to do a post-processing based on a probability map, but as Uwe mentioned, right, like you only have 32 rays in this case, and you cannot hope with 32 rays to capture the complete intricacies of every detail in the membrane, right? So you always have this, like a little bit of slack that could happen if you really want to precisely outline a membrane. You probably should use StarDist for the interior and use that as your seed region for a little bit of watershed growing at the end, right? And the watershed growing, you can actually use what I will speak about now, which is like a probability map that is only foreground background, right? Because in crowd cells environment, like the hardest part is always to get the seeds correctly and not miss seeds and not get falsely merged seeds. But the growing actually typically is mostly a fine or like an easier part than finding correct seeds. Okay.
I have a quick question about the number of rays. So I know, like, okay, so I know in the notebooks, there's why you can choose the number of rays that gives you like the shape of your data. So I would understand that if it's like, if from like, let's say 64 above gives you your shape of arrays, it should be like all the number above that. But sometimes when I'm looking at the graph in there, that it looks like it's going down when it's like above 96. Is that like,
okay, no, it should not be, no, that there's no, like logically, it should never go down. If you increase the rays, the number, yeah. That is interesting. We should have a look together because that is certainly a bug.
Okay, yeah, because it goes down like when I'm training 3D models.
No, but
you mean the accuracy? No, you mean the best accuracy you can hope for, like this initial step where we compute for the number of chosen rays, what your ground truth reconstruction accuracy would look like, right?
Yes.
Yes.
Yeah.
Yeah, that, I mean, it could fluctuate a little bit because of course, like the number, the actually points that is chosen is different, but it should not, I mean, perceptually, it should not go down like by a lot, right? It should wiggle a little bit, but this is just due to the different changes of points that you use to discretize, but it should not like drop 10% or so.
Okay. But in 3D, right, the points change every time, right? If you increase the number of points, you get different positions.
Yeah, that's what I mean.
2D,
exactly.
That's not
so
the
that's
case,
it,
right? In 2D, you always keep the points from last time and add more, so it shouldn't go down, I think. But in 3D, it can happen through these discretization, so it can fluctuate a little bit.
It can fluctuate, but it should not really like 10%, 20% like loss of accuracy.
No, it's no. It's just a little bit.
Okay, well, you can probably then show it later in the breakout.
Yeah, I can. I can show you. Yeah.
Maybe if I can jump in, just one last little question here for the number of rays. So usually in these images, when it's nuclei, your number of, let's say the number of pixels in a nuclei is relatively small. So this 32 ray will give you a quite good accuracy of the reconstruction, right? But now if I'm segmenting a larger object, which still can follow your approach, my density, my pixel density is much higher. because this object is larger, should I then go for a larger number of rays to try to reconstruct better the edges, or should I downsample my image, try to fit in what a nuclear would look like?
So typically you will always get the same relative resolution. So what you will miss on the edges will always, the proportion of that compared to the complete nuclei will more or less stay constant, right? It's just the absolute number of pixels that you will miss will change, right? And now is the question if that is really something that is super important to your pipeline or not, right? If it's really necessary to get like 5% accuracy of the area, that's super important for a biological question. Then of course, then StarDist at all can be used only as the first step anyhow, right? So you would use that as like this plus minus 10% outline of the cell. So you'd use that as something, maybe get the probability mask of foreground background and use and expand your slightly a bit according to this probability map. Right? So that would be my recommendation for these kind of situations. Any other questions?
To ask for the scaling of the 3D distance map. So you do not scale the input image or your mask, but you scale, so to say, the vector of the case.
So we don't scale. I don't understand what do you mean by scaling the vectors.
So if you have so XY is typically square-like and high resolution, and in Z you have
okay, yeah. No, I understand what you mean by scaling. Yes, what we do is actually, so what we typically don't scale anything of the stacks. Maybe we should, but no, we don't do it now. So we keep the raw images. So you predict on the raw images. And if you imagine like your raw images have like 5- or 6-fold less resolution in Z, or sampled less fine in Z, like 6-fold, then if you have spheres, perfectly spheres, then they would look like prolate compressed objects, right? And of course, if you then just use normal rays that are equally distributed on a normal sphere, they would miss a lot on the side, right? And that's what I mean by anisotropy. So what we do is actually we make sure that if you would have a sphere in your Platonic world that is then prolated in your compressed stack, then your points would be equally distributed on this prolated sphere, right? So we kind of distribute more rays on those regions that are hard to reconstruct in your stacks, if it's anisotropic, right? So this is already kind of factored in, and that's what I meant by that. Yeah. So we change actually the directions according to the anisotropy.
Okay.
And this anisotropy is actually, when you use the notebooks for that, it's automatically detected because that can be computed from your ground truth, right? If it sees in your ground truth annotations a lot of compressed objects, it will detect that and will automatically set the anisotropy to this value, right? So that's something it's actually, you don't have to take care of that, but it's just like that's happening under the hood. Okay.
Yeah, in one sentence, the distribution of the rays is selected such that the objects in your ground truth can be well represented. Yeah. That's also what's shown in first notebook. It shows the reconstruction accuracy with and without anisotropy. And you see that you need fewer rays to get a better, good reconstruction when you take the anisotropy of your objects into account.
So if we have a low resolution image, is it worth upscaling it before we run StarDist, or will it be fine if the nucleus is like 10 pixels wide?
That should be fine. So I would not upscale. The only reason or the only use cases we had where we saw improved results is if you have images that are really like 10-fold less resolved in Z. So everything would be super thin. That is very challenging. And they're upsampling in Z and a little bit downsampling in XY such that it's more or less a little bit more isotropic than before. This kind of seems to help.
I mean, like about like 2D images, do you think it matters?
Yeah, 3D. Or you mean 2D?
Yeah, I'm talking about 2D.
No, 2D. Don't do anything to 2D. I wouldn't.
Okay.
Unless the objects are like 200 pixels, right? Okay. They should as mentioned, they should like objects should fit into the field of view of your network. And in the notebooks, we will automatically detect if that's the case. And if you see in the notebooks that your object size is too large for the field of view, that's actually like, it will print that if it happens. What you can do then, for instance, you can then like downscale your images. But this is only if your images are 10, if your objects are 10 pixels, that's no problem. It's more like if your objects are suddenly 100 pixels or 200 pixels or something extremely large, and downscaling actually would still, like visually, it's still, you can still tell what is an object by downscaling it. then you should do that. But that's something we check in the notebooks.
Thanks.
Mm-hmm. Any other question? Okay. So this is just an example of like some nuclei in C. elegans, right? And where it works in the same way. And what we recently added to it, and we just mentioned it, that instead of only presenting or classifying or detecting objects, so only predicting those star convex shapes per pixel, you can now additionally, and this is in a developmental branch, but soon will be in the main repo, you can additionally kind of label each instance with a different set of discrete classes. So for instance, in this case, we not only have like these instances labeled, but each of those instances has one of two classes, belongs to one or two classes. This is just a synthetic data set where you see like we just assigned every cell that has a texture to the blue class and everything that is constant to the orange class. But of course, this could be like cancer versus non-cancer cells or something like this, right? And how you actually do that under the hood is like the normal branch or the normal network that only predicts those distances and this object probabilities. You just attach from a layer that is not the last, but a little bit before last. So one that already has extracts a lot of features and semantic representations of your input image. You just add another head, like a prediction head that predicts those class probabilities, right? So that's very easy to do under the hood, but sometimes helps a lot when you want to outline those cells. And from that, you then get not only those instances, but as well for every of those instances, you get one of the classes that you predefined, right? So they can use that to classify cells instead of only detect them. And of course, it as well gives you a foreground background probability for all the classes. So if you're interested in, this is still a little bit, I mean, we haven't really tied that up in a super easy to use interface, but if you're interested in and want to hack a little bit, of course you can now use this prediction, because this prediction is essentially a foreground background prediction per class. You could just merge that and use that as your way to expand the original StarDist markers according to this class in your watershed, for instance, right? So that's something that is probably very simple to do if you really want to have that outline be pushed to the border precisely. Another addition that we do is efficient inference for large images.
Before you go to that, maybe just this object classification, it's not ready yet. It's not part of the regular notebook, so it's in development, but you can, so if you install StarDist in Python from the GitHub repository, there's a way to do this. So you can ask us about this. You could play around with this kind of like an early access if you want to be a bit of a beta tester. Otherwise, this will eventually find its way into the normal StarDist package that you install in Python. And hopefully then also trickle down to the Fiji plugin eventually.
It currently lives in its own branch, yeah. But this branch, you can install it with pip if you're a Python user and just play around. There's a notebook detailing that, so you can try that out. So, And the other one is like inference or prediction on very large images. And what we do there is just to show you there's some tricks that we implemented to make sure that it runs on very large images. And very large in this case means like 20-ish gigabyte for 2D images, which is not extremely large, but for our purposes, it's considered large already. And what we do is essentially we do a tile prediction. So we split everything into tiles that fit in the GPU memory. And then we do some, and this is just to show you this succession of runtime and memory as it goes down. And what we do is we put it into smaller tiles that we use for the GPU and larger tiles that we use for the NMS. And then we predict it on overlapping blocks and stitch then back together everything that is predicted twice, right? Which is not trivial, but can be of course be done in some way by doing this NMS and this overlap block 2 and stitching them back and make sure that you don't get kind of strange looking artifacts at the border of those blocks. So it's a lot of little boring work, but at the end, if you look at the graphs, the runtime, so the red one is after doing all the tricks, the runtime goes down from quadratic to linear. This is a log plot, and the memory almost stays below the input size, because we can actually as well predict on something what is called a ZAR array. And the ZAR array is just an image that is not loaded to disk, loaded in memory, but it's actually just presented as a folder structure on your hard drive. So it even works on effectively arbitrary sized images if you have the time for that. So just to give you a ballpark, so for like a 20 gigabyte, 18 gigabyte image, 100K by 100K whole slide image that we tested it in pathology, it's roughly 50 minutes. So and it gets at the end 8 million nuclei from 62 million candidates. So this is feasible, still takes a little bit of time, but it should scale almost linear and should actually work for these type of images. What will you see?
Just
yes.
one comment. So bottom line is like if you have some really big data, so if you previously ran into like memory issues, if you ran it on big images, then this big thing will take care of that. So in principle, you can run it on arbitrarily sized images that don't even have to fit in RAM on one machine. So what's not parallelized yet is if you have like terabyte size images, you cannot use several workers at the moment to run the prediction in parallel, but we might add that in the future. But just in case, if you're worrying that it will not scale to your data, it will. Yeah, so you don't have to worry about, unless you're really like you having like a terabyte size thing every week or so, then it's maybe not ready yet for you. But otherwise, you shouldn't have to worry that it. So it will work with your data typically.
But this edition is in GitHub master branch, or is it...
This is in the dev, like the predict big.
The big thing. Okay, so the big code is in the main repository. So if you install the latest version, it has all the big functionality. The notebook that is explaining how this works is still in the dev branch, but you can still use it today. It's just I can give you the notebook.
Mm-hmm.
Yeah.
So it all works in the main StarDist that you install with pip with the current regular version. It's just we haven't advertised how this works at the moment. That's more like the notebooks and explanation how this works is not really in the main thing, but it doesn't mean it doesn't work in the main.
Great, thank you.
I mean, if you actually encountered that for that problem and want to get around this, we just ask us in the breakout rooms and we'll show you how to do it. Okay. Okay. One thing that you will see in the notebooks and the tutorial notebooks is something called data augmentation. And this is just a way to artificially increase your annotations that you made of your ground truth objects and present a network with different slightly changed geometric versions of that, copies during training, to just artificially increase your data set. And this could be, for instance, like flips and rotations, right? This is what people typically do, or elastic deformation. So you would, both the input as the output would be automatically deformed a little bit, and or added, or noise is added to the input, of course, not to the output. And this is kind of, you can do that within the notebooks with a specific function that you can then add your own augmentations if you want to just artificially increase your training data and make the network more robust to unseen data, right? So this is something, if you have a limited amount of training data, is always advised to do. And just a short comment on annotation software, which of course that's the input to the method, right? So you have to annotate your stacks. And in 2D, the situation looks pretty good. So you have a lot of tools that are available for doing this annotation. So there's Fiji, where you can just draw the voice into an image directly, or you can use something that is called LabKit. So LabKit is developed by people from Dresden too. And it's actually used as a random forest classifier, so something like Elastic, but it has a way to annotate images for doing actually the classification. And it's a very nice way to annotate images. So it has a big data viewer backend, and it provides like tools for annotating 2D and 3D images. So we kind of abuse it as our recommendations for 2D and 3D annotations, because it's really nicely done and it ties very well into Fiji. And for 2D images, there's as well QPath, probably some of you already have used that, which provides a very nice way to draw very effectively a large set of annotations in 2D images. And there's some, you always have to export those images into a label mask, so into a dense array or a dense stack of label images, or an integer image where each individual pixel carries one value. And we provide some scripts to do that for each of those packages. So LabKit and Fiji does it. LabKit does it automatically. For Fiji, you have to use a script, and for QPath too. In 3D, the situation is a bit more involved, because you essentially have to go through every slice and label every object that is the same with the same number, right? So typically the workflow is you go through an image, that's Z by Z slice by Z slice, and you just concentrate on one nucleus or one instance of the thing and label it with the same color until you reach the bottom. And then you go to the next one, which is of course sounds very painful and is very painful, but that's the way it is currently. And for that, I would suggest using LabKit, because it does it in 3D as well as in 2D. And if you have very, very large volumes and want to have a little bit more of an advanced Pro tool, there's as well Pantera from the Sulfate lab, which is a very advanced tool that provides interpolation of labels. So you don't have to label everything, but you can actually interpolate between different Z spacings that are apart, certain Z planes, and the labeling is automatically filled in, which is quite nice if you don't want to really go through every Z plane. And yes, and if you want to use, yes.
So for the 3D annotation with LabKit, so let's say a cell is clear on some stacks and it's not as clear on other stacks, do you have to annotate like all the stacks, or can just focus on the clear ones?
So typically this depends. If you want to segment all of them, so you should annotate what you want to have segmented at the end, right? So this is the first thing. As before, if you have blurry objects that you're not sure, if you annotate them, they will be segmented. If you don't need them to be segmented, then you should not annotate them, right? So you should annotate every object that you then at the location step or detection step you want to get. And if this includes hard to see, hard visible objects, then you should annotate them. If you can get away with losing them, you should not annotate them, right? So this has to correspond all the time.
No, I mean, when you say you have to annotate with the same number, like one cell, the same number from stack to stack. So it's the same cell, but it's visible. It's visible in certain stacks and it's not in other stacks.
Then you should annotate it through, right? Because implicitly, you know that it's one instance, right? You just can see it as well, right? But if it's really fluctuating between these slides, you should annotate it continuously, right? Yes. Okay. So another thing is that
sorry, about the 2D or 3D segmentation. So you mentioned the pain of annotating 3D data. I recently took a course about deep learning in 3D image segmentation, and a lot of people there actually took the approach of using 2D nets and then finding clever ways of combining the output of those into 3D, just because it's much easier to get annotated data. Do you have a comparison between like StarDist 2D applied to 3D images along different axes and your 3D StarDist models?
So we haven't done that, and it's very interesting. Yeah, what you would do is you predict every Z-plane, right? And then you get a lot of candidates that belong to the same objects in different Z-planes, and then you would have to find a way to merge them, which you can already do in some sense. So we could already do it, but we never did it because we don't have time for that. But it's actually super interesting to see that. Yeah. So you could, in principle, you could already do it now by yourself, because we have part of StarDist is a matching submodule that does matching of ground truth objects in 2D or 3D objects with the predictions. And of course, you could use that to match the output of different Z slices, and it will tell you which one overlap. And then you would just identify them and go greedy Z by plane and just match overlapping or just fuse overlapping matched cell instances, right? So you could already do that if you want to try around with that. We just haven't, we haven't packaged it into a nice API that is just a single line, but you can already do that. But it's an interesting question, yeah.
But yeah, and sometimes it's a combinatorial thing, right? Sometimes you have multiple things that may match together, right? And what do you do then? So if you do it in the greedy way, like slice by slice, then it might not be optimal, right? It's a bit of a tricky problem in some cases, maybe. So like when we did a session on Monday, like in some sense, right? If you imagine every slice is a detection, and then you treat the Z slices as time, then use a tracking framework to combine the different slices. In some sense, that could probably work, right? If you take the detections and then load it into a tracking framework, maybe that will actually do some nice job of merging the different segmentation from the different slices. Yeah. Could be, I don't know, we never tried it.
Just use TrackMate, yeah.
So it really depends on some data. It's probably the greedy approach works beautifully and you get around the painful 3D annotation. But since this wasn't like a clear-cut, obvious thing to do, and it depends on some data, it will probably fail. That's why we haven't done anything with it, and also due to lack of time. Yeah, but doesn't mean it's not a good idea.
Actually, I think it's a very good idea, but yeah, we should probably make it more but we haven't implemented it in a straightforward way to use. Yeah, you would have to find your way around it on the Python level, which would be interesting. So. Okay, so again, everything is the bare bone of everything is a Python module that can be installable via pip. So pip install stardust should give you everything. You will see that in the notebooks later on. And then if you have a 2D model that you trained or you want to use a pre-trained one, for 2D, we currently have a Fiji plugin where you can use either pre-trained models or 2D, your own trained 2D models. And this works as well on CPU as well on GPU, so it should work on your computer. And pre-trained models for 2D, we already have for fluorescence microscopy. So this Kaggle challenge data set where you have nuclei and fluorescent nuclei labeled. And for RGB images, H&E staining and histopathology, where we use the data set as publicly available. And these two models are already in the Fiji plugin. And they can be used in this way, relatively painlessly, where you can select different models, and this includes models that you have trained yourself. Of course, this only works in 2D so far, so that's a bit of a current issue that we need to expand, but if you have a 2D problem, that's a nice way to test out if your models work on unseen data. Okay, again, there's a GitHub page, and if there are questions further down the line in the next weeks or next days, go to the forum with the tag StarDist, and we should get a notification. And yes, these are some practical considerations for training your own models, and I think these are just some best practices that we find. So always get a simple baseline. If you see something, if you have a detection or segmentation task, always check what is a simple thresholding give you. So just do not get too excited too early. It's always good about, so this is the biggest thing about actually both image restoration and StarDist object detection, like the training data acquisition and getting good training data. This is like 70% of all the work. It's just a common source of error if you have ambiguous cases that you just forgot to label in some case, it's not registered. And all these are common sources of mistakes. So make sure that this is in place, and always beware of outliers. So normalize your data correctly. And this was all demonstrated in the notebooks on practical cases, and registration for image-to-image restorations. And if you see something, if you want to try out, so this is something for StarDist 2D and 3D that we would always recommend before like getting excited or getting too excited and just trying to label in 3 or 4 weeks the complete stacks that you have. First, just do it on a single crop that you do, and just try to what is called overfit. So you really want to overfit a model on a single stack and just see, does it work at all, right? And of course, it won't generalize to new stacks, but it's just like it's a baseline, or it's the maximal performance that you can hope to achieve. And if this is failing, then there's some other problems that you have to take care first, right? Maybe data annotation or image normalization or something is weird, but always overfit on a small data set first to see if it's worth annotating more, right? What else I would recommend for StarDist annotation specifically in 3D is maybe first use a simple pixel-wise classifier to get some rough outlines and do a watershed on that, and then curate those as your input ground truth masks, because really annotating 3D is extremely painful. And this is kind of a shortcut that we see now in a lot of projects people took. And it actually works quite well, because splitting cells that are falsely merged, it's way easier than really kind of annotating every cell. If your data set consists of 80% easy cases, then it's not worth annotating all of the easy cases when you then don't have any motivation left when it comes to the hard cases. And that's just my recommendation.
Another question on that. So what do you think of the approach of basically using a classical imperfect pipeline to annotate a lot of volumes, and then training a model on that and hoping basically that the errors are random and you get better model performance? Have you tried something like that?
This you can do. I mean, this is an approach that in theory should work. The only problem is that the aspect that you actually correctly mentioned, that you have to make sure that your segmentation mistakes are randomly distributed, right? And typically, especially the case for crowded objects, they always fail consistently at those cases that are crowded, right? And in principle, if you have if your watershed, for instance, if you have a lot of objects that are touching a little bit, and your watershed will always fuse them, because you just get one seed for two objects that are fusing, and they're still a dent, so it still look like two objects. What StarDist is in principle cannot do something that is bean shaped, right? Or that has kind of two spheres that are touching. So it's kind of forced to predict two objects there. So it could work even in these cases, but it's not guaranteed that it doesn't make like a single object in the middle instead of two that you actually want. So this is something that is hard to say in general. It could work, but if you really typically it works for those cases that are easy to start with anyhow. And the hard ones are those that a normal classical pipeline would just miss. And these are the cases you're actually interested in. These are the borders that cannot be inferred with classical approaches. And these borders are then not borders anymore in your annotations, right? But it's worth a try, because it's essentially something that you can get very quickly. Yeah, sure.
But yeah, we had that at some workshop. Somebody used the watershed and used it for training StarDist, and StarDist actually fixed some of the mistakes in the watershed. So it can really work. It really, as Martin said, right? If you have two shapes that are like this, the watershed merge, which are not star convex, then StarDist cannot make this mistake by design, right? So you get this split automatically. So it's worth a try. And yeah, starting with some imperfect annotation, cleaning them up, taking the imperfect annotation, training, cleaning up, and iterating on that could be an effective way to get high quality annotations.
I just want to quickly show you how to annotate with LabKit in 3D, if that's interesting. And this is all done in Fiji. So if you go here, this is the update site. And oops, that is, if I go to update, you will see it's something that I already installed. And you can actually find it in the update side of Fiji under LabKit. So just check this box and install it. And I loaded a stack, and these are cell nuclei that we see before from C. elegans. So the input stack would be just a C stack of that. So it's a 205 by 101 by 140-ish. So this is roughly a size that I would always recommend annotating first. So you see some of those nuclei. And what you want to do is, that's a little trick. You want to first make sure that your Z, you want to map Z to time. And I will explain that later, because that makes annotating a little bit easier. And then you would just go to LabKit, you open it in LabKit. And here you see it's Big Data Viewer backend, so you can scroll through different Z positions. And here you see the labels that you have. And as I mentioned, LabKit is originally designed for foreground-background annotation. So the initial default classes are background and foreground, but we want more of those classes, so we remove them and just add label by label. And here you have like a tool set where you can paint stuff, for instance, like a brush of a given size. And what you would do then, of course, is, for instance, you focus on one nuclei, paint this nuclei, go to the next slice, paint it, go to the next slice, paint it until it's almost gone. Okay. So here you see. Okay, so I think this one is over. Then you go to the next label. And that should be a little bit more precise, maybe. Go here. Like this. And then, of course, the easy step, do that for all the nuclei. and then you can export it by just save labeling, and you can save it as a TIFF image. I just say it's the test stack mask.tiff. And then if I reopen that, I get what is the input for StarDist for that stack, which is just an where is it? It's hidden. There should be some labels here. Yeah, they're hidden somewhere. Here. Okay. So you get a labeled integer image where each cell has a different label. So this has 1, this has 2, and of course there would be all the others if you follow through. And that would essentially be the input, the corresponding mask to this TIFF input. And that's all you need then for StarDist. And you would put that then in your local drive or on your Google Drive, and then you can use the notebooks based on that data, right? And of course, it's not only 2 cells, but you would have to label all of them, which is of course a little bit the painful part, but LabKit is very nice for doing this on small specs because it has nice tools to, here you, for instance, can erase stuff. If you're not happy with that, you can correct it. So it's very nice. Okay. Questions so far?
I have a question. Yes. If you have a membrane staining, for example, in this case, so would you draw the membrane or would you draw the
No, you would draw the interior.
Okay. But you would consider it as a background then or as a foreground?
So you don't, that's what I'm saying. Yeah, that's why I mentioned it. So you would don't do foreground background. So you would really do every instance, every interior of the membrane would have a different label. So you would create a new label, add label for every of those instances. And you would just label the interior, right?
Okay.
Any other questions?
So why do you recommend to use LabKit if Pantera doesn't require to do this on each stack?
Yes.
Yeah, so I LabKit, not Pantera.
No, I just typically LabKit is a little bit easier to install because Pantera you need to have a Conda environment. It is a little bit more involved to install. It's way more powerful than LabKit, but LabKit comes with Fiji, so that's always our first first baseline for people to use because you can just install it. There's an update site. You click a button. It's quite easy to install and use. But if you're familiar with Pantera and you have large data sets and you want to have a more powerful environment for annotating, then Pantera is there.
Yeah, and Pantera,
LabKit is
as Martin mentioned, right? You can annotate one slice in the beginning and one slice in the end, and will interpolate all the slices in between, for example. And you can also get the 3D visualization of your annotations in Pantera.
Yeah, it's really cool.
But yeah, so it was made to proofread large connectomics, segmentation, and so on. It's meant for EM data and so on. That's where it comes from. So it's very powerful, but it can be, or at least it was a bit difficult to install. I'm not sure how it is these days. But yeah, if you do this a lot, this annotation, or have dedicated people who check annotations, or you want to fix annotations later on, and you do this a lot, then it's really worth a look to go for Pantera. Okay, so I'm not sure, are you already overwhelmed with everything that Martin presented? Because I'm going to talk about some quite technical topics. So this is mostly meant for people who want to maybe develop their own neural network and want to use some of the tools and infrastructure that we have in CSBDeep especially. And so I'm trying to understand how much time I should spend on this. So, or just for people in facilities who maybe want to distribute pre-trained models and such things. And as I said this in the beginning, this is new content. So this is maybe a bit rough still. So and my plan is to go through this rather quickly. And if anything sounds interesting to you, then you can ask me later about it. But of course, feel free to stop me while I talk about this. Otherwise, I will really keep this short. Then we have more time. So you can, in these small groups, in these breakout rooms, you can talk about specific topics, right? We separate people into groups. And then you can talk among yourselves, and we also join and then help you with your problems. I think that's probably what most people would like to do in the remaining time. Then if that sounds good to you, can you nod or raise your hand or something? Okay, so like I said, I'll go really, this notebook is in the GitHub repo. You can run it on Colab, so it's if you want to do it later. I'll go through it really quickly because it's covering really more advanced topics geared towards developers. Okay, so we have these two libraries that Martin and I develop and maintain, that's CSBDeep and StarDist. And all of this infrastructure stuff really lives in CSBDeep, but it's also used inside of StarDist. So all of the stuff I'm going to talk about is kind of like the foundation of how this works under the hood in our libraries. Okay, Yeah, so the main topics I'm going to talk about is that our library works with TensorFlow 1 and 2. So how do we do that, and how can you do that? Like write code that works with both library versions. Then what kind of neural network blocks and nets we provide. Then we have the concept of a base config and base model. That is kind of like the base class for our models, and how you could use those to like write your own models, and why this could be maybe interesting to you. How what we do for pre-trained models, and how you could do that. For example, you made a cool model, you work in a facility, you want to share it with your users, how you could do that if you use a model in CSBDeep or StarDist. I have a fairly involved example, like how we putting all of these things together, but let's see if we can get through that. And then also Martin already talked about how you can break a big image into tiles, process them separately. So we have some utility functions for that if you want to use them. Okay, so let's get started. Okay, so we have some basic imports for this. This is not really important, importing standard stuff like NumPy for arrays and plotting and so on. We in our library we have this function called keras import, and this will import the right keras whether you're working with TensorFlow 1 and 2, and the API in keras and changed. So keras is now part of TensorFlow since version 2, so everything changed a bit. This is important for developers, and if you want to actually write code that works with both versions of TensorFlow, this could be useful to you. So this will import the right keras depending on whether you use TensorFlow 1 or 2. Okay, then what kind of neural networks can you do? So we have these functions called common unit and custom unit. There's just in common unit you have fewer parameters exposed. But you already heard that unit is this kind of like this workhorse in like the life sciences, like deep learning in life sciences. A lot of is based on units. I mean, they're more recent models, but units is typically quite robust, and it's also powers a lot of things in CSBDeep and StarDist still. I mean, they're newer models out there, and they might work a bit better, but they're sometimes a bit more fiddly and so on. But it's yeah, and so, for example, you can make a model like this. You just give it certain parameters. You can like take a look and see all the things it can do, right? And you have a little bit of documentation. If you're a developer, you can look this up. And then this model of summary is a way in keras how you can print kind of like a textual summary of a model, in case you haven't seen that, but it shows you the model you created. Okay, and then of course you can mix and match keras. So there's the keras input layer. You can mix it with, for example, a ResNet block that's in our library and combine it to make a model like this, for example. Okay, so like I said, I'll go through it really briefly just to show you what's available, and I will not stop unless you ask any questions. Okay, but this, like I said, this is developer focused. Okay, then really important, everything we do is based on this base config and base model. So by default, a base config, and I should preface this by saying this is what we needed so far. So in case you want to use this and you might have different needs, we could also like incorporate those or think about it. So like all our models have the notion of axes. So when it's all of the models take an image as an input, and this is the axes for the neural network model. This is like the semantic metadata. So for example, this is a 2D image, first like the height, then the width, then the channel dimension. This is what a neural network expects. That's represented then in the model. So we know a number of channel in, out, like is it 2D or 3D. And then we have certain things, checkpoints. When we train a neural network, we automatically save the weights during training to disk. So we always remember the best weights according to the validation loss. And also we save the weights that are currently the neural network, so at the current epoch, and also after training. And by default, we use these names: weights best, weights now, weights last. This is like some standard we always use, and this happens automatically. There's some convenience functions for that. You can extend this base config rather easily, and then add your own parameters like this. You can add it, and then you can see you created your own config. It has the name you give the class. And with this config,
type that you created, you can, for example, now create a new model type that derived from the space model that we have, and you have to provide at least two things. You have to say, what is the class of the config that this model expects? So in this case, it's my config. And you have a function that builds the keras neural network. In this case, I'm just not doing anything, but here you would define this. And then you can then create a model like this. So you create a new model of the class my model. You provide the config parameter, a name, and a base directory where this model will be created. And then you can see in the output here, so now we have a model that by default, the axis that it takes is this. It creates an output of the same axis. It lives in this directory, and it has this config. So this, and it will automatically persist the config to disk. So you see, when we look what's inside the model folder, it has written the config as a JSON file, has saved it to disk automatically. And you can look at the JSON, it has all the parameters that are shown here. And you can then later load this model from disk by just providing none for the config file. And that means you should load this model from disk. So this is what you do typically after you train the model. It will load the configuration and all the trained model files automatically for you, that we can then use it for prediction. See, like you even get this warning, right? Couldn't find any network weights, because it will automatically look for any weights, and it will load the weights according to the best validation of automatically for you. And yeah, and there are lots of other things in this, other helper functions that I don't have time to go into, but some of them I shall show below. One of them is the registry for pre-trained models. So if you may have used StarDist and used the pre-trained model for StarDist, like if it's in Fiji, you get this drop-down list. In Python, you can just call this function called from pre-trained. And this will, if you don't give it any argument, it will just show you all the pre-trained models that are known for this. And you can then instantiate it by either giving the name or an alias of one of these pre-trained models. It will load the model automatically from the internet and cache it locally and so on. And then you see the StarDist model, when you print it, it looks a lot like this model we defined ourselves. So the syntax and the structure is all the same. But the config for the StarDist model is quite a bit bigger here. It's like all the parameters it needs. But you couldn't actually call the same function on this my model class that we just defined, right? You can also call from pre-trained on this. It just doesn't know about any pre-trained model yet. But you can then like register your own pre-trained model by giving it a name and a URL of the zip file of this model folder, and then also the hash of the zip file, so it knows if it's already downloaded the correct file. And you can also give it aliases. And when you do that and you call the from pre-trained function, you see now this new model class that you just created now knows about pre-trained models. It has a name and these two aliases for it, and you could instantiate the model that you uploaded somewhere in this way. If you give it then the name, for example, here, my model, it would then instantiate the model. Of course, this URL doesn't exist, and it will like throw in a mistake here now, right? Because it's just a fake URL I put here. Okay. So far about pre-trained models. Just this could be useful for people who work in facilities and want to distribute these models. But you can also add new pre-trained models to StarDist or things like that. Here's like a bigger example that uses, so Martin talked about these semantic segmentation, and also this way, for example, you predict the boundary of an object as a separate class, so to avoid touching nuclei. So this is just a little demo here I made. So I don't want to go into the details. You can also look this up later. So I defined some helper functions just to avoid lengthy code. So it's like I crop all images to the same shape, convert them to the necessary format that we want to predict, define a loss function, and have a simple data generator that does some flipping and rotations and so on for this data. It's not as important. Then the data we use is just the data we use for StarDist in the example notebooks, which I load, crop to the same size, and so on, normalize. And then it's just like you have an input image. This is like the labeling we have, and this is now the that the segmentation mask I created, that I want to train a neural network to predict this. So I want to train a neural network that takes the input image and predicts the segmentation mask, okay? And the segmentation mask, the boundary of each nuclei is a different class to have a chance to separate those nuclei that are touching, okay? It's a common trick that people use. Okay, and then this is kind of like the part I want to show is, so I want to now define a model for this task. So I make a new config, I call it now seg-config for segmentation, doesn't matter, just pick any name. And I can add any new parameters I want. Here I just added a simple argument, kind of like this UNet has a certain depth, and I can pass this as an argument. Then I have to say, what's the configuration class that I need here? This function build like constructs the keras neural network, and I'm using this common UNet function that's in CSBDeep. And you pass any arguments. So some things are hard coded here, but you could put them into the config, right? And just read it from the config instead. And this will instantiate the model. Then there's a function that prepares the neural network for training. So in keras, you have to compile it. And so you have to say, like, how do I optimize the neural network weights? What is my loss function? And so on. This is like standard keras stuff. The nice thing that the space model does for you is like it automatically creates callbacks that automatically save the weights during training for you. So some nice convenience functions. And then there's this thing called TensorBoard that allows you to monitor the training progress. And we have a special TensorBoard thing in the CSBDeep library that actually automatically shows all input and output images of a neural network in TensorBoard. So you can easily and quickly monitor how well the model is training. And I'll show you this in a second. And then, so when you train it, you prepare this model for training, then you call this fit function on this keras model. It's the standard way of doing neural network training in keras. And then at the end, you call this convenience function that's also bundled training finished, and this will automatically save the weights and then load the weights at the best validation loss automatically for you. So you get a nicely trained model at the end. And yeah, the predict function, I'm gonna talk about a little bit later when it makes sense. But let's for now say, okay, we made this model, we created a config, we instantiated the model and so on. We can take a look at the model it uses, doesn't matter so much. And now then we can train the model. So like as Martin mentioned, right, we are taking the data that we loaded, and in this case, I split it up into training and validation data. You could also split it into a test set, but here in this case, I only did train and validation. And then I'm calling the training on the model. And then you see it's training for a while. So I did this a bit earlier, right? But then while it's doing that, we can take a look at this TensorBoard thing. So the TensorBoard, in case you haven't seen it, is a tool that's it's shipped with TensorFlow, and it shows you to like see this is your loss function during training. You can see how it changes over time. It's very useful to debug, and also shows it separately for your training and validation images. So you can see that the loss goes down, which is to be expected, but you can also find out when it's overfitting and so on. But so this care TensorBoard thing that we have made specifically for images. So if you're using that tool, then you can automatically, let me make this a bit smaller so it fits. So automatically it will show you all, these are like just 3 of the validation images, will show you the input to the neural network, the prediction of the neural network, and the ground truth, so what the network is supposed to predict. So for example, you can then see over time, so in the beginning, the neural network predicted this after 1 epoch, and then later, so it's getting better over time. And that, so this training I stopped. Sometimes it's a bit buggy, this TensorBoard thing, but so you see that now the bluish outline is like starting to appear. And what this is super useful when you start a training and you made some mistake, you can immediately see right away when your training goes wrong, because you can, after like the first epoch, immediately see how the predictions of the model change over time for your images. So in case you're a developer, or you can also do this for StarDist and CSBDeep. This TensorBoard thing is always logged to this, and you can always monitor the progress of your training in this TensorBoard and look how the prediction for 3 validation images changes over time. Okay. But it's also, if you write your own custom models, you can also do that. So let me make this bigger again. Okay. then after training, you can see there are a bunch of files in our model folder. It saved the weights at the best validation loss. It saved the weights at the very end of neural network training. And it saved these weird files here that are for the tensor board. So the tensor board that can read these files and show you these graphs and these images. And then, yeah, so then everything is persisted in this model folder, and you can load your model at any time later by giving this none for this config. And that means I'm loading everything from disk for this neural network. I load the config and all the neural network weights. So you see this status message that's loaded the weights from this weights best file. And then, so how do you predict for new images after training? So you have direct access to the keras neural network, and you can call the predict function on that. But this will only work if your images are properly shaped and already normalized and so on. So this unit, for example, expects that your image is divisible by like 2, 4, 8, or 16 or something like this, because it will downsample your image and upsample it again. And if your image is not divisible by a certain power of 2, then it will not work. And what you do for that is actually pad the image or crop the image beforehand, so you don't have this problem. And then, for example, we have tools to do that. That's why I'm scrolling up now and go to this predict function that I defined earlier. So we have a bunch of tools. We have these normalizer and resizer concepts in CSBDeep. And here, for example, I'm assuming that images are already normalized. That's why normalizer equals none. But we have this thing called the pad and crop resizer that takes the input image, pads it, and later removes what was padded from the prediction. So this is kind of like, and also when you call a predict function, you tell what are the axes of the image that I'm inputting. So for example, let's say the model trains, the trained model expects that the channel is always at the end, but then you load an image from disk where the channels are in the front. So you can put in the axis of the image that you want to predict on, and will automatically internally move the image axis around such that everything works for you. And this works throughout CSBDeep and StarDist. So it's the necessary step. So we take the axes, we check that everything works out. Yeah, then we normalize the image. We resize it by padding. So the before method is before prediction. It will pad the image. Then it will run the prediction with the actual neural network, and then later the resizer will call this after function. In this case, it will crop the image. But you can write your own resizer that do whatever you want with the data. Okay, and then, so if you have, for example, an image that has an odd shape like this 223 by 223, which does not work with this unit, and you try to run it with this neural network directly, you get this big error message, something that the sizes are not right. But if you now call with this function that we just defined, then it will pad the image to a good size and then crop away what we padded. Okay, and this, for example, is now what the prediction would look like after I trained this just for a little bit. So the prediction looks crappy. It's just, yeah, it's just what the output is at this moment. And one last thing is that if you assume you have a really big image, this is not really big, but that's just an example I'm using here. Then we have this thing called a tile iterator in CSBDeep that chops up your images into certain blocks. So it's maybe easiest to show in here. So each block will always start at a multiple of a block size. In this case, the block size is 8. And in these, for example, this UNet neural network will give a different result if you shift the image 1 pixel over. So that's why it's important to always start as a certain, if you shift the image or pad it, it should only be a certain increments of a certain block size. And inside CSBDeep and StarDist, we do everything automatically for you under the hood, but you can use the same tools if you wanted to define your own models. And then you can define how much should these blocks overlap. And this is always given in multiple of this block size. Everything is given in multiple of this block sizes. And in this case, I think it's like 5. So it's like 40 pixels is the overlap here. So it's, and then how many tiles do we want? So we want 3 in height and 5 in width. And you see then it will also know what to crop away if you want to save it later. So you see if you concatenated all the magenta regions together, you get back the original image. All the other stuff is padding that's necessary for the neural network to do its job, because you always need some context around it. So you run the neural network on these individual blocks, then you crop away this thing and save only this part in magenta to the final output, and then you get the same result as if you were to predict on the original image or directly. And that's the stuff that happens under the hood in CSBDeep and StarDist for you. If you input this parameter n_tiles. So this is just shown in here. Yeah, so it's as simple as calling this tile iterator with all these parameters, the number, the block sizes, and so on. And then this line is the crucial bit. It's kind of like you process your tile, you crop away the non-magenta region, and it knows where to write it back to the original array, to the correct location. That's all you have to do. So you don't have to worry about how do I make these blocks, where should I start and end, and so on. This is all taken care of for you. Okay, with that, I'm done with this whirlwind overview tour of CSBDeep.
Uwe?
Yes.
Is there a kind of rule of thumb about the overlap of the blocks?
You mean how big it should be?
Yes, how big the overlap has to be.
Yes, this is here. Here I use an arbitrary value, but in general, it depends on your neural network, on the architecture of your neural network. For UNets, and you can actually calculate that, right? If you use a standard UNet model, and we have helper functions that pre-computed how much overlap you needed for UNets. So we use that in CSBDeep. Otherwise, you can also determine this computationally by, for example, making a constant image and like a 0 image, and you put 1 pixel in the center, and then you can compute gradients, and you see kind of like what's the influence reach of certain. And we do this in StarDist actually automatically. So if you have a StarDist model, the first time you use one of these tiling things, it will compute for you how big is this area, region of influence, and will then adjust the block sizes automatically for you. So there's lots of things happening under the hood in order to get a correct result.
Okay.
But yeah, there's a function called, I forget the name now, but there are functions in StarDist and CSBDeep in the KER networks that tell you how much the block sizes should be. And but yeah, this is something you have to, this is neural network dependent. So it's not easy to give a answer that works in general, right? But you can always determine it empirically by just running a computation.
Okay, thanks.
And that's what we do in StarDist, because it's not that as easy anymore. Okay. And with that, I'm done with this. So are there any more questions? I know this is like super brief, and I just wanted to, this is the first time I'm like representing these more internals of CSBDeep. And this is more like an overview. If anything sounds interesting to you, you can take a look, dig a bit deeper. And a lot of the stuff is not as well documented as all the user-facing functions, right? So everything in the notebook and that people typically use, it's much more well documented. But this is meant as a starting point. If anything of this sounds interesting to you, then yeah, ask us about it. And also if something is not quite right, but you would like to use it, then we can also think about making it more general. Okay. And the final thing I would briefly show is Martin mentioned that we have this prediction on big data. And there's also a Jupyter notebook for that. So in case you're interested in running StarDist on big images, we have a tutorial notebook for this. It's not advertised yet, and it's also in the developer branch of the GitHub repo. But yeah, it should work fine. I'm not sure the API is going to change. So I think this is all good. And this is just showing like we have one of like, this is a 30K by 30K image roughly. And it's just showing like progressively zoom in to just show how big it is. And then how would you run StarDist on this? And yeah, it's talking a bit about normalization and how you could normalize this without making a copy. It's not so important here, but the normal way of running StarDist would be this. There's like this predict instances function, but this could be prohibitive in terms of memory and as Martin showed in the slide. So there's this method called predict instances big now that takes some additional parameters which are documented here. Most notably is now there's a block size. So you take this block out, and the neural network prediction and the non-maximum suppression happens in this one block, and then you have a new block that's shifted, and the overlap is chosen in such a way that every predicted shape is either fully contained in one of the blocks. If you can guarantee that, then you can then reconstruct everything, and this works. And this is some additional parameters you have to provide in addition to. So this parameter is for the tiling of the model for GPU processing, and this block thing is for like the outer blocks. It's a bit complicated for the outer blocks for which you do the non-maximum suppression. So if you don't have huge data and you don't run into these issues, don't use this function. That's really for people who cannot do the non-maximum suppression all together because it doesn't fit in memory typically. And then you can run this, and with some tricks, you can actually see the blocks where it was processed from and so on. And you can also do this if you, so this typically we work with NumPy arrays, which live in memory. So you have to load your image into memory. But you can also use this with ZAR, which is an array library that allows you to work with images that are only loaded on demand. So you can, this is just showing how you would, for example, write this image that we have in memory here to a ZAR array, and then use this for input and also writing to a ZAR array, the output of that. So this also works. So this means you don't even have to load the input into memory anymore. Okay, I think this was all very quick, I know, but yeah, I'm happy to go into more detail if people are really interested in some of these things. Otherwise, I think it's important that you play with the stuff that interests you as well, and then we can help you if you have any questions with that. Okay, so I'm done.
It was really nice to see what's under the hood and the things you can tweak. Easy to repeat or to think about yourself, but for me, but it was great. Thanks.
But yeah, some of the stuff is just we use it internally, but somebody else could use it potentially as well. But we've never advertised it because, I mean, this was now this tutorial was the occasion to actually make a notebook like this, because it's always work to even like show how this stuff works. But this stuff I showed in this developer focus thing is not as well documented because it all takes a lot of time to make it nice and user friendly.
Yeah, actually, I have a question. Coming back to my point before, when you're a user, but you have like a very small annotated data set, I was thinking it could be like interesting to use a model which is already trained on a very large data set, and then basically use this model as a starting point with your limited, basically annotated data set to train it and get a good result. So I understand this is possible within that framework, but I know from the deep learning literature, there are many ways to do that things. Some people will just basically take the pre-trained model and load the weights and use these weights as a starting point of the training. I think this is called fine-tuning. And some people will have a more sophisticated approach where they will freeze some of the layers of the pre-trained network and then, for example, just basically keep unfrozen some of the other layers to retrain on the new annotated data set. So all these approaches possible within your framework, and which one would you recommend for the kind of usage I have described?
Yeah, so pre-training definitely makes sense. And ideally, we would have a notebook that also shows you how to do this, right? And it's definitely possible because you have full access to this keras. So keras is this neural network library that we use under the hood. And there you have full control to freeze layers and so on, right? You can change everything you want. So this model structure, this base model that I described, is just like an outer wrapper over the standard keras model that you have full control over. So you could mix that. And I've tried it once to like fine tune a network, and so what's the best way of doing this? It's like a bit of an open question, right? This has to be tried. And so far, it's just something we haven't done yet. But this would be interesting to see how, like the different ways of fine tuning, which one works best and so on. I would also like to know that. Like which layers to freeze in the beginning and so on, right? This is something to be tried. And it would be nice if somebody tries.
So people actually did that. So Olivier from Bori from EPFL last time mentioned that that what they used at the end was they tried fine-tuning, and that worked okay. But what worked the best was just using the original data set it was trained and just put your own images to it, which is a bit, it sounds very not the correct way.
This works if your data is similar enough, I think, right? I think it's not always the best way, but it could be.
Yeah, my point is not just that, like, typically what works best is very much depending on your data. And we typically here try to, everything that is a publicly exposed function should be pretty foolproof in deep learning nomenclature. So, and this would be something where you really you have to experiment a little bit. Of course, you could just do it yourself, but including that would mean you would have to find reasonable, you would actually have to carry out a lot of research just to find reasonable defaults, right? And that's why it's not part of it, yeah. But it's totally true. I mean, you can just fine tune it with a small learning rate, for instance. That's what people do.
Yeah,
and
but
it's totally feasible.
Will just involve one of the pre-trained model you have already. And then basically just train it with your small data set with a very low learning rate and see if it performs well.
It's one line of code extra. It's just before you train a model, load from pre-trained. That's it.
Okay. There might be some details. Maybe we need to make a copy with that. And there's some little technical issues, but it's nothing complicated.
Yeah.
Okay, thank you.
So what are we going to do now is go together through one of the workflows that we prepared, and all of them can be found at the GitHub at StarDist minus I2K here that I showed you, and you hopefully all have access to. And at the bottom, there are some workflows that we prepared that are runnable in Google Colab. And we will now, based on the initial poll that we did, most of you are interested in 3D object segmentation. So I would propose what we do is the following. We all go together through one workflow and StarDist 3D together, and you follow along. And if there are any questions in intermediate, we're going to try to solve them live as a whole. Additionally, for those who already have a super specific use case and want to just hack around and get our feedback on that, might be 2D image restoration, 2D StarDist, 3D, or those advanced topics that Uwe just mentioned, we created a breakout room that you should all be able to join. It's located in the tab breakout rooms, which is, I think you should all have in the Zoom interface, you should have a breakout room that currently no one is in. It's called room 1. But Uwe is going to be in room 1 during the next half an hour or 40 minutes that we do the tutorial notebook in the main room. So if you already have some super specific use case, you know what to do, you want to do something that is not 3D StarDist or just want to be quicker with everything, just go into that room. Uwe will be there and can guide you through your problems that you might have, might be data annotation, all these kind of things that are not in line with just a regular 3D StarDist workflow. Just go into this room 1, and I would propose Uwe is just there and waits for.
Okay, I'll go over to room 1, and if you want to, we can just talk in room 1. Okay.
I mean, you can go back and forth. I'll try to make the run through in the main room as slow as possible, that it's easy for people to follow along. But if you already know what you're doing, it's probably best to go into room number 1, and you can cycle back and forth. I see already you're allowed to join. So, okay. For the rest of you.
What's the link for room number 1? I can't find it.
There's just one room. If you go to breakout rooms, there should be just, I mean, if you want to, I can assign you, but I want you to be able to go back and forth.
Yeah.
So if you go to breakout rooms in the Zoom interface.
Oh, I see. Okay.
And there should be just one, hopefully, and should be able to join. And for the rest of you, just go to the GitHub page. And then we're going to open up the third example, which is StarDist 3D, based on the initial poll. Okay. Okay. Everyone is on the GitHub page, and then open the Colab. And if you click on this badge, well, this notebook should open. And it should look like this for everyone. Okay. So this is like a example notebook for the 3D workflow, and it automatically will download some data that we created for demonstration purposes. So don't worry about that. You could, of course, adapt it to your own needs by uploading data on Google Drive. But if you want to do that, go to the room with Uwe. He can guide you through that. Everybody sees that page. And if not, please let me know.
I don't yet for the moment.
Okay. So it's on the bottom of our GitHub repo. Here, open and collab. And you have to sign into your Google account for that.
I'm good.
Okay. Everyone else good? Okay, good. So I'll make that full size. Okay. So everybody who hasn't used Jupyter Notebooks before? Raise of hands. Okay, so just for those who haven't used them, so Jupyter Notebooks are just a sequence of Python steps. So each cell contains code, and you can run it by just going to a cell that is runnable, and it looks like this, and it has a little play button on the left. And you just go in succession and follow each code cell and run it, and you'll see how it goes. But essentially pressing this button will run the code that is contained in this code cell. And if you click on those little buttons to the left, it will expand and show you what part of the code actually each cell corresponds to, right? And for everyone else, we just start at installation and preparation. This is normally, if you don't use Colab, this is you do once on your local machine, and it's essentially just this install command pip install and pip install stardust. So if you run that, you can ignore the first one. It probably will tell you that it's not authored by Google, but we say run anyway. And this will install stardust, it will install CSBDeep and keras and all the dependency. And that should hopefully be, in my case, it was already finished because I just run that notebook before, but it should be done in like half a minute or so on your machine. Okay. So I will try to just go along, and if something on your side is not up to the speed, just I rely on you interrupting me, okay? Otherwise, I will assume I'm too slow. Okay, so after installing it, we will just import some packages that we need for image loading, for plotting, and from Stardust itself. And this pertains to ray definitions and the model definition itself, and you can just run those cells, which essentially just imports some stuff that we need. And generally, each workflow consists of 3 main steps, and we label them 1, 2, 3, data, training, and prediction. So in the first step, you just load your data, look at it, see if everything is correct. So you do some sanity checks, see what is the input, what is the label mask, did you name your images correctly that they correspond? In the second step, the actual training process, you will read those stacks and the model will be built and trained. And finally, you will use the trained model on stacks you haven't seen. So this is a general, like the workflow for each pipeline for our cases, in this particular case for Stardust 3D. And we just go through each of those steps together, one after another. So the first one is the data part where we read and download the data. And we're first going to download some data that we prepared for the demonstration purposes. And you just hit that code cell, it will download the data. I already did that, so I have nothing to download, but for you, it might take some seconds to download everything. And in that data set, we prepared some stacks that consist of 3D input images, raw images, and the corresponding label mask that you would get after annotating, right? And if you run the next code cell, it just lists the data structure or the folder structure that everyone should now have on his Google Colab drive. It's called data, and it's split into a train and a test set. And each one of them contains subfolders that are labeled images and masks. And in the images, there's just a list of images of raw images. And in the masks, there's corresponding images that have the same name of the masks. So that's the generic storage or layout that we use for StarDist. And for instance, we can peek into if you look what's inside of data. Now, for instance, in the train folder and images.
Excuse me, I got
a list of images.
I got some errors trying to download it.
Okay, can you share your screen?
Yes.
Everyone else managed to download the data?
Okay. I can't share the screen probably because
I'm probably sharing. Now it should be. Now you should be able. Yeah.
Do you see it?
Oh, you have to run the first code cell where we import everything. So just download and extract. Yeah. So you did the pip install exactly.
Yeah, I did. Yeah.
And if you go to the next one, no, don't redo that. Exactly this one. No, below. If you go down. Exactly this one.
Okay. So
because this imports this function. Yes.
Yeah.
So that's why in notebooks you always have to be, every code cell has to be run in succession, and yeah, and it should hopefully download now.
Okay, thanks.
Sure. Okay. And if you just look into side, for instance, the images folder, it looks like this. There are 26 images that are called stack 000 and so forth. These contain the raw images. And if you look into the masks folder, masks, it has corresponding stacks that are named identical, but they contain now the label mask. And we're gonna look at the labels a little bit later on. So here we're just gonna load this data. We just read all the file names.
By label, you mean this integer
exactly.
Integer numbers for
the annotations, exactly. And we have a look at later on in one of the cells. This is just reading the image paths and loading them into disk. And we're going to use just some small data, a subset of that for visualizing. This is going to read the images. And this is what I mentioned before. This will calculate the anisotropy of your annotations automatically. And it will calculate this empirical anisotropy. And we're going to have a look at an example image later on. And I will mention that probably then down there. But it will compute an automatic anisotropy along Z, that the Z pixel size is twice the one from XY. In Python, everything is always ordered Z, Y, X. That's why the first number corresponds to the pixel size in Z. That's the pixel size in Y, and that is first in Z, in Y, and in X. And now we're just going to plot some example images. That's probably easier to see. And this is this code cell. And this is an XY and a XZ slice of the raw data. And this is of the corresponding label masks, right? And the anisotropy that I mentioned is, if you see at the axial slice, like the pixel size in Z is twice as large as in XY. That's why they look like prolate objects, right? They're a little bit stretched. And that's actually inferred automatically from your annotations, okay? Does everybody sees that?
Yeah, and this when you say that it's infer, it's because you assume that the objects, the volume has to be isotropic, so it should look like spheres, or how do you calculate?
No, it's just exactly like internally, it just uses that to distribute those rays way better than it would be if it would be spheres, right? Because they're not spheres, they're kind of compressed objects, so it kind of internally uses that information to distribute those rays better, right? And you see this factor 2 is essentially the factor that you had to apply to make it isotropic. That's essentially this anisotropy.
Yeah, so basically if I would take that one on the bottom and multiply by 2, let's say, then it becomes more roundish.
Exactly. If you have images that are isotropic everywhere, the code cell above would just tell you anisotropy is 1, 1, 1. So no anisotropy found, right? And if it's worse, then it's a respective factor more, right? Okay. The next thing is we're going to do is we first try to see whether our labels and objects that we want to classify for, are they actually well representable by the star convex shape priors, right? So what we're going to do is we kind of compute the rays for the ground truth and see if we can reconstitute the ground truth if we would have perfect prediction, right? And that's just here. That's just a function that does that. And we're going to change the number of rays a little bit just to see if there's a sweet spot that we should use. So we're going to use 8 rays, 16 rays, 32, and so forth. And this will take some time. And it will compute automatically what is your accuracy if you would do that, right, on your ground truth images. And we will do it in 2 cases. First, if we ignore anisotropy, so we effectively assume it's isotropic, which is not. And the second one will use the correctly calculated anisotropy, right? And in your cases, this is all automatically done, so you don't have to put anything manually there. But this is just for seeing the effect of having anisotropic distribution of rays. And then we're going to plot the accuracy for both cases. And here you see, it's a bit hard to see, but on the x-axis is the number of rays that we used. And on the y-axis is the accuracy. And with accuracy, that's the mean intersection over union. And you see actually it dips a little bit. That's interesting, yeah. But you see a difference between the anisotropic and the isotropic case. And typically, so the sweet spot is typically in this order. So you don't want to use too many because then you probably have discretizations. The distribution of rays is a little bit different. And you don't want to use only 16 rays, right? So the sweet spot is in this middle. And that's typically our default anyhow. So with 96 rays, you're pretty fine, right? You're in this region. You should just avoid being in this region. But generally, this is just for demonstration anyhow. You wouldn't use 8 rays. That's clearly not enough, right? And that's just showing that this is the case. Okay. Again, this is just a sanity check. You don't have to do that for your own data. You can just right away go into training, but that's always good to have a sanity check if everything works. And here, in the next step, we're just going to reconstruct based on those number of rays. Yes.
The accuracy, on what is it based? Is it pixel by pixel? So that means there's 100% overlap of the ground truth and the mask, or is it, oh, you found 50 and I found 50, or I found 50?
So actually, we have our own. So if you look into this function, what we compute is we use a function called matching dataset, and this matching dataset computes accuracy of detections and overlap. So it computes a huge array of possible metrics. And what we're going to use is the mean true score. And the mean true score is the IOU, the average IOU of every matched object. So if this metric is 0.9, it means on average, you get 90% overlap for each object, right? And that is the y-axis here. And this next cell is just plotting some of those reconstructions, right? Again, this is just for illustration. You don't have to do that for your own data, but it's just nice to see. So here you see the XY slice of the reconstruction when you do use different rays, right? And clearly, if you use just 8 rays, your polyhedra are super, super coarse and clearly not a good approximation. But then if you go down, it looks more and more like it should look, right? And later on, we're going to use 96 rays. So this is like a good default for these kind of shapes. Okay. Everyone with me so far? Good. Okay, then we come to the more interesting part, which is the actual training. So now we have checked that everything is correct. Our images and the labels are corresponding and they can be reconstructed, at least to a certain accuracy with StarDist. And now instead of just using 2 images, we load the whole data set of 27 files, so 27 images. We're going to read it. By the way, this will automatically check if your input image is how many channels your input image has. So if your input image has 3 or 4 channels, this will work too. And it will set it automatically based on the shape of your array. And the next step is that you want to normalize your images. So typically your images contain values between 0 or 500 pixel intensity, whatever your camera and camera exposure and your laser power is used. And the network always expects that your input is kind of normalized in some way. And what we do, we kind of squish the range of images into a range of 0 to 1 according to percentiles. So we calculate percentiles, and this is all done in one function that we provide. And in this case here, we normalize images based on the 1st and the 99.8th percentile, such that we kind of have a robust min and max estimation, because sometimes you have hot pixels and you don't want to use the max of a hot pixel to squash your data set to to a super tiny range. And we do a little bit more post-processing on your ground truth annotation, because sometimes if you label with some tools, you sometimes have a single pixel in the middle of an annotation object that you forgot to label. And for StarDist, this is a very bad thing, because it would think there is a border and your distances will be calculated with respect to the interior pixel that you forgot to annotate, right? And that's super bad. And on the other hand, it's super easy to miss, right? Because in 3D, sometimes you miss that. And this is kind of filling those holes, right? So this is just a sanity check that normally should not be necessary, but it's always better to be safe than sorry.
Martin?
Yes.
You mentioned before multi-channel images. So are you training StarDist on all 4 channels? I mean, you had 4 nuclear channel, for instance, with 4 different nuclear dyes. You could say take all 4. Do you need to make a max projection between the 4 channels, or what is it that you...
So the input is always, if you give 3 or 4 channels as an input, it will use them independently and it will normalize them independently. So it will use all the information in all channels, but without projecting them. If you want to exclude certain channels and say, don't look at this channel, then just don't provide that channel, right? So by default, it will use everything that you give it, and it will actually learn like how to mix them properly, such that the final segmentation is the best, right? Okay, everyone fine? Good. It's super important that you say so if you're not fine, because otherwise we're gonna lose people, and that's not what I want. Okay. So the next thing we do is what I already spoke about, is that we're gonna split our 27 stacks into a train and a validation set. And the train set will be used during training, and on the validation set, it will be constantly evaluated if we're not overfitting, right?
Sorry, Martin.
Yes.
Something popped into my head. What happens if at some point you do this normalization also during analysis, I guess? And what if you are in a bad lock where you some sort of crop an area where there is no nucleus?
Yes, that is something that you should avoid.
Right. Okay.
Yes.
So there should always be an object somehow.
There should always be an object, yeah. I mean, you can, that is like, if you want to run the notebook as is, that's the case. You should always have something that that doesn't break the normalization. Of course, this is Python code, right? You can do whatever you want here. You can just do divide by 1,000, right? If that's and then it would be fine for background stacks, right? But that's actually a super important point, because otherwise background patches, if you just have noise and you normalize noise, like everything will be popped up. It will still be fine. It won't make mistakes there. But then later on, it might be a problematic part. If you normalize your whole image, the background would be normalized differently, right?
Right. So it might be good that perhaps in these conditions, normalize like using larger areas and then use small patches just for the analysis, let's say. Something
exactly.
Like that. Okay.
Cool. Okay. So we split our 27 images stacks that we have in total into 23 training and 4 validation. And it will warn you if you not have enough training data if you have a single stack only. It's quite helpful. You should change that. Okay, and 27 is a good, it's a good number for 3D. So these stacks are, I think, 256 times 256 times 128 or so. And then again, we just plot some labels. In this case, the first, and that's, as we've seen before, these are just the labels that you have seen before, okay? Now, having done that and checked everything is right, so the left and the right side correspond, we actually go to constructing a network, constructing a StarDist model. And we do that by providing, so every model has attached to it a configuration object in which all the details that you typically you don't need to change them, but you can if you want to, right? And this is all hidden inside of this config 3D object. And if you do this, it just prints the documentation of that. And in the documentation, you see all the things that you can change, but should not change, okay? But these are like, just that you know, everything, almost everything can be changed if you see it. And this is, of course, later if you do training and sometimes you have a gut feeling certain things are not wrong and you ask other people or you ask us, then we can pinpoint you to, oh, maybe change this a little bit to make it even better, right? So this is the purpose of that. But you see, for instance, like the architecture, what filters you use, how deep is your architecture, what backbone you use, the number of training steps, the number of training epochs, the batch size, and all these kind of things that you don't have to understand now, but later on you probably will, if you do that a lot of times. These all can be tweaked within this configuration object. And then we're going to calculate again the extents. And we see, yeah, now we have 2 and 1, 1. So this is our extent or anisotropy that we empirically calculate. And now we define the actual configuration object. Okay. So the only thing that you want to change is the grid parameter that I will speak in a second. So the first one is just we define the number of rays and we set it to 96 because we found out that's a reasonable default. And then we're going to set the grid parameter, and the grid parameter is a list of 3 numbers that are integer that by default are 1, 1, 1. And they just say, so if you have, imagine you have a lot of large stacks and you don't want to predict at every pixel the distances because you essentially just want to find out where the objects and where's the outline. So it makes sense to predict only every second pixel, right? Just to save memory, GPU memory, to make training faster, to make inference faster and all that. And that is this grid parameter. If you set it to 2, 2, 2, for instance, it will, along each dimension, it will just predict every second. It will still see the complete normally sampled input image, just your final prediction will using every second pixel. So this makes sense if your nuclei or your objects that you want to segment is larger than 10, 20 pixels or so, right? If your objects are very super, super tiny, like 5 pixels, of course, you would not set that to something that just predicts every second pixel. But we make a default that if the anisotropy is larger than 1.5, just use every second pixel, right? And this is just for demonstration purposes. And then you construct rays. This is just our definition how we distribute rays with a golden spiral. And then you construct a configuration object that gets as an input the number of rays that we use, 96, the grid, the anisotropy, the number of channels that are automatically computed to be 1. And then this is another. Excuse me. Yes.
Could you maybe explain just one more time what anisotropy is? Because I got confused with the grid values.
Yes. Yeah. That's actually true. Yeah. It's a bit. So the anisotropy, if you look here at the XZ, at the axial slices of the input data, okay, the anisotropy is just like assume your input would be actually perfect spheres, and you see this image. What pixel size this corresponds to result into raw stacks that are looking like this, right? And here you would get this image, actually, if your pixel size in Z would be twice as large as in XY. They would look like this if you open them in Fiji, right, without rescaling. And that's just the anisotropy. The anisotropy just says how much more they are squeezed into each dimension with respect to a normal sphere. And in this case, they're squeezed twofold along this dimension. So the anisotropy factor along this dimension, which is the first dimension, is 2, and the others is 1, right?
Okay.
So the anisotropy factor means what do I have to scale your raw images? With what factor do I have to scale everything along each dimension such that they will be spherical at the end? And that's essentially, I have to scale it up twofold along this dimension and leave it in the others. And then they would become perfect spheres, right? That's the meaning of it. Okay.
Okay. So if the anisotropy is big enough, then we can use the
Exactly. So actually what it does is if the anisotropy is larger than a certain amount, so if they're squashed along the Z dimension, then you should be fine. You should make your prediction finely sampled to not miss one of the Z planes that are the important ones. But if they're actually quite spherical, you can subsample faster, right? So you can subsample more. And that's all what is, and that's automatically calculated, but it's just for context, that's what's happening under the hood.
I understand, thank you.
And the only thing that you probably need to change too is what is called the patch size. So what StarDist does actually, it uses your training images and it will crop out small 3D patches that fit into GPU memory automatically, will augment them and will randomly crop those patches during training from different portions of your sample. And the size of that patches is kind of depending on, or it's actually limited by the GPU memory that you have, right? If you have a GPU with small memory, you need to decrease those patch size. In an ideal world, you would just use your raw images, the full raw images, but they never fit on a GPU in 3D, right? So that's why you have to cut out small patches. And this has to be made as large as possible that it fits into your GPU, because this patch size is essentially what your network will see during training. It will see patches of that size. And the big thing, or one mistake that can happen is that your cells are larger than this patch size, because then your network only sees portions of cells. And that's very, very bad, because it has then no notion of where's the border if it just sees like the interior of a cell. So you always have to make sure that this patch size is larger than the nuclei or the cells that you want to segment, right? And if this is not the case, and you cannot make it larger because your GPU memory is limited, then I would suggest just downscaling your images to start with, right? Such that they don't become larger than 96 pixels, okay? Which for our case is not true, but it might happen. And it will warn you if this is the case, okay? So we print a warning that's hopefully, yeah. Okay, any questions regarding the construction of the network? So again, you don't have to understand anything in detail too much, but it's just an option that you can choose. So having done this configuration object, we just construct a StarDist 3D model with that configuration, and we point it to a folder where we should store all the models. In our case, it's just relative folder called models, and we give it a name, in this case, just StarDist, and everything will be saved to that folder, every intermediate results. And finally, at the end, and we will come to that later, if training is done, and if you want to export it to Fiji or so, everything lives in that folder. So the model architecture, the config, everything is stored there. So you don't have to then worry about that anymore. So if you run that, it will just build that model and save the configuration to disk and prepare everything. And then what I mentioned before, it will do an additional check based on the model's receptive field. So each CNN has, based on the kernel sizes and how many max pooling steps it does, it has a limited receptive field. And your nuclei annotation mass should fit into that receptive field. And it will just check that. And if this is the case, if it prints out the warning, you can increase the grid size or you can downscale your stacks, okay? So it will tell you, okay, your median object size is 10 pixels in Z, 2020 in XY, and the network field of view is 1730/30. And this, of course, depends on the network architecture, right? So what kind of backbone you use, because it depends on how many filters, how many max pooling steps you can do. And just to show you something that you can change easily, if this happens, that your objects are larger than 30 pixels in XY, which is often the case. So the easiest thing to do is just to change the backbone. So currently we use as a backbone a ResNet, and a ResNet is powerful, but has a relatively small field of view. So you would, for instance, just use here backbone, change that to something else. And in this case, for instance, we have a UNet backbone. A UNet backbone has a larger field of view. I just rerun that. Okay. Equals. So now I set a configuration object with a different backbone that has a larger field of view. If we now rebuild the model, it will warn me that it's already existing. The folder is already existing, but it will override it. And now we see the field of view of the network has changed, right? It's a little bit larger. And of course, you can make the network even bigger, then it gets larger and larger, but that's more of a detail for if you see there's a problem, okay? So far, so good? Okay, good. The next step is that although we have 27 stacks, it's always good to do some basic augmentations that are already mentioned in the main overview lecture. And these are, yes.
Just a quick question. Mm-hmm. Do you recommend UNet over ResNet or ResNet over UNet, or?
So most cases, it does a similar job. It's only the only case where I would recommend the UNet is if your field of view is a little bit larger than if you see that warning essentially.
Thank you.
So yeah, but it should pop up. So you should realize that this is the case. And okay, so the next step is augmentation. So although we have 27 stacks or 23 for training, it's always advisable to do some super basic augmentations. And this includes just flipping your images, like just X as a line flip. So you just rotate it or you mirror it, because this should not, your output of your images or like your segmentation should not depend on whether it's flipped or not. And the important part for 3D is that in 3D, it's not really true that your images are symmetric under arbitrary flips, specifically for the Z dimension, right? Typically, you cannot tell whether an image is flipped in X and Y just by looking at it from XY. But if you look from the side, you can tell, because the one closer to the camera will be the one with the higher SNR, right? You always have this depth-depending loss of SNR. So you should not flip it along Z, because that would not lead to images that look identical to original images, right? So that's why this is done automatically here. So it will only do the flips along X and Y. So you give it here. Okay. So here, this is the parameter that is constructed. So we're only going to use the axis, the first and the second axis within Python is XY. Okay. And what we do additionally is we just do random intensity changes. So we just multiply each intensity input image. We multiply with a random number between 0.6 and 2. So we kind of scale it a little bit and add a little bit of an offset, of a random offset. And this is just a single value for the whole pixel. So it just shifts a little bit and makes it more robust to normalization mistakes. Or if you don't normalize it properly during inference later on, it's way more robust if you just shift the intensities a little bit during training. Okay. So these are two basic augmentations that you can always do. There are way more fancier augmentation methods that you can additionally apply to it. For instance, elastic augmentation or addition of noise or scaling and rotations. But they can all be tied into this function that we provide. So you define a function that takes as an input image and the label mask, and it does some augmentation and spits back the augmented images. And here you can do whatever you want. And by default, we just do random flip rods and random intensity changes. But here this is an entry point that you can use for whatever augmentation you're going to use, right? And this function then later will be given to the training function of the model. So it's going to use this function. And just as a sanity check, this again, it's nothing to do with deep learning per se, but with the data. It's super important to just plot some augmented images, because if you do a mistake here, oh, I have to run it first. If you do a mistake in the augmentation, for instance, you just flip your input, but forgot to flip your output, it will still train, but the results will be really, really bad, right? And so you have to make sure that this is not happening. And for that reason, we just plot some examples of the augmented images, and you see some of them are brighter, some of them are flipped, and you should always make sure that they're still corresponding after the augmentation, right? Which is the case here. Okay. Any questions? Okay. Then comes the exciting part, the model training. And we always, so what we do during model training, we not only train the model, but successively during training, there will be intermediate output. So for instance, the loss over time and intermediate results will be written to something that is called a TensorBoard log that you can then look in the browser, and I will explain that later on. But these logs live in a file called, in a subfolder log. So we're going to remove them if they exist. It doesn't exist now, but if you rerun it, you would see other old results, and you don't want that. So this is just this little cell. So it removes that. It's not existing, so it doesn't hurt. And then we're going to load the TensorBoard extension, and I will show that again later. So bear with me for a moment. And the TensorBoard just allows you to look at model progress, and it will pop up a little GUI here that will be then populated later when we do the training. This should change. Okay, so we're going to leave that for now. And then we're going to do the actual training. Okay. And the actual training, especially for 3D models, can take quite a while to really converge it to something good. So typically this takes 5 for up to 12, 14 hours. So we typically do that overnight for the final run. But for the first quick test, you don't need to have pixel-wise perfect result at the end. So we're going to do just a quick demo first, which means we just use some epochs. We train it just for a short amount of time that fits into a tutorial. And that's why this flag, we have this flag. So if you want to have a longer training, just set that to false and it will train longer, right? So that's the only difference. So we have a quick demo, we set it to true. So we actually execute this branch. And here in this branch, the actually model training function is executed. And the parameters of this function are just the training pairs, X and Y training that are the input. We're going to provide the validation data. So this is the data that we're going to evaluate after each epoch, our accuracy. We're going to provide the augmenter function that we define. So this will be called within the training script all the time. This can be none if you don't want to do any augmentation, but we define some of them. So we're going to use that. And we just use 5 epochs. And an epoch is one step over the whole 23 training stacks. And typically you want to use 100 epochs but that would take 10 hours. So we're going to use 5. And this is the steps per epoch. This is just how many stacks you want to visit each epoch. And we're going to set that to 10. Typically, that's 100. So don't worry about those numbers. By default, they're set to something sensible. But here, we just voluntarily make that smaller to make it feasible. And then we hit the play button. It should train. And it tells you it's only for a quick demonstration. And if your output should now be the individual epochs and the different losses during each epoch. And this should now progress over time. So this is the number of steps per epoch. We're still in the first epoch, and you should see your training loss, which is the first number here, which is 2. That should decrease, decrease, and decrease. Of course, it's very, very not really convenient to look at this loss curves, although I can, yeah, it has a certain amount of suspense just looking at this loss curves for hours, but that's not a good way to do that. So that's why we use TensorBoard under the hood to depict the progress of the training. And if you now go up again, so this will run now for 2-3 minutes. And in the meantime, we can go up again to TensorBoard. And if you refresh it on the right side, there's a button refresh. It should now be populated with values. And hopefully everybody sees that. And there are 2 important aspects that you can watch, and they're located to the left at the menu. It's called scalars and images.
Martin?
Yes.
Sorry, can you explain again how you went to this these graphs? I missed that.
Did you, so you run that code cell?
Yeah.
The extension reloading? Okay, sure.
So,
or just share your screen. Yeah, share your screen.
No, sorry, I got muted. Okay, I share my screen. Yeah. So I am here.
Mm-hmm.
I have to run
yeah,
this.
That's Don't rerun it. Oh.
Ah, shit! Sorry, I'm so sorry. I didn't hear you in time.
No, it might still, yeah, it might still work. So the training is going on. Okay, if you go up again, and now go to the refresh button that is on the right, this little circle, the spinning, the circle with an arrow within this TensorBoard GUI.
This one. Okay.
Yeah, hooray.
Cool, cool. Thank you. I have to stop sharing somehow.
I think I can force you to stop. Yeah.
Maybe there's a command line flag to automatic. So there's a way to automatically update every like 15 seconds, I think is the smallest interval you can choose. So when you hit the little gear icon, there's an auto update. Maybe you can actually do this in a command line so you don't have to hit that button every time you want to see updates.
Should already do that every 30 seconds, but
the default is that it's not on, which is stupid, but whatever. Yeah.
So if you keep on hitting reload and then press, so what you see in this graph it's called scalars, and that's just a fancy way of saying these are the loss curves, the different losses that get minimized during training. And stardust is composed of a lot of losses, one pertaining to the object probability, one pertaining to the distance calculation. And the final loss that matters is called epoch loss. And if you click on this button here, it will kind of refresh that. So you should see those two curves up until the final epoch, which is just 5, but this would go to 100 or so. And you see 2 curves. One is blue, one is red. And the meaning of that is explained here on the left side. You see the blue one has train at the end, the red one has validation at the end, and they correspond to the loss curves for the training and the validation set, okay? So the blue one is for training, and that should always go down. It should always perfectly follow almost an exponential decay. And the validation one should go down too, should follow the training curve. And at one point, it will deviate and just not go higher or go up again. And if this goes up again, that's a case of overfitting, right? So you want to make sure that your validation loss is decreasing all the time too, right? And we check this moment where it happens that your validation loss is best, and this will be then used for the final model storage. So the model will be saved at the moment when the validation loss is the best, okay? And here, as I mentioned, there are different loss aspects. So for instance, there's the probability loss, and there's the distance loss. So you can see all those loss functions independently, but this is more like if you really want to debug things. The main important point is you should go to the main epoch_loss, and you should see both curves going down. If you don't see that, something is wrong, okay? And another cool thing is under the images tab, if you go there, you can see the intermediate results. Again, you have three things that you see. You see the input, your current prediction output, and the annotation masks. The first one is the inputs, and this is just the max projection or slice. I think the middle slice of it, yeah. So this is your input, slices through your input validation data. And this is your target, okay? And these are the intermediate results. And you see,
that's a bit, so yeah. Target means this is the automatically created from your instances. This is the distance map that the model is, some of them, right? So that's what the neural network is supposed to do with your data. That's created from your annotations automatically, so internally. And the colors mean this for like the distances for 3 different of these radial directions. So it's a bit otherwise it's confusing to understand what it means, but you can use this to see how well it's doing.
Mm-hmm. And you see, like after 5 epochs, it barely got to know what is a nuclei-ish. So it hasn't done really anything, which is expected after 5 epochs because we just trained it for 2 minutes. You would train that for 100 epochs or so 3-4 hours. And then you would get actually a nice loss curve. I will do that at the end if time permits and just let it run and show you that. And after a while, so what we do for the quick demo, so if you set it to false, it will do the actual run, and this will take just longer than a tutorial. What we do after the training, we just load one of the models that we just did train longer just for demonstration purposes, okay? So the model is now pre-trained and that our prediction makes sense.
Yeah, I think the 3D demo model is exactly what would happen if you run this,
yes,
like
that is,
without the quick demo mode.
If you set quick demo to false, it will just train for 3 hours or so. Everything will be fine. And that's the model that is loaded if you set from pre-train. That's exactly our demo model. It's coming from that notebook. Yeah. Okay. So at the end, after all this is gone, you should have seen that the loss curves all went down. Your intermediate results look reasonable. And what is then done at the end after you trained your actual model, you always have certain thresholds that you have to either set manually or automatically for the thresholding of your object probabilities. That means how many objects you get at the end and of the potential overlap that you would allow. So StarDist allows for overlap, and it will only suppress polygons that have a certain amount of overlap. So if you want to have no overlap, you can set it to a small number. Or if you want to have heavy overlap, you set it to a large number. But this is a value, this threshold is not a priori meaningful. So we kind of do a quick sweep over optimizing this threshold based on your ground truth. So this will automatically done. It will find the best thresholds. And this is done in this little code cell. So we optimize those 2 thresholds that are actually buried inside of StarDist. And it will do that automatically based on getting the best accuracy based on your validation set. Okay. And this may take some time. So you see it will try certain maximum suppression thresholds. And here is the updated best accuracy that it can get.
But Martin?
Yes.
So we discussed before that we don't use overlapping nuclei for the training.
Yes.
So it's seen an overlapping nuclei. So how does it evaluate this this threshold?
Yeah, this is the overlap for the NMS, so for the like rejection of polygon candidates, right? Because sometimes you want to allow close by nuclei that still then are touching, but there are objects that are predicted are overlapping a little bit before you render them to the final image. And sometimes you want to allow that, and it's not clear what is the optimal threshold for this object, for this NMS step. So this is for the internal object candidate rejection, right? There's nothing, it has not so much to do with the actually overlap that you get at the end, that's true.
And yeah, by the way, so this is getting good thresholds on average. So if you care in your application that you will never want any overlap whatsoever, then you should set this overlap threshold to 0, for example. So this is like just, if you use these thresholds, you get closest to what your annotation would have been on the validation data. That's what these thresholds mean.
And these are just the defaults for it. So we will later see in the prediction scripts and in Fiji, you can manually do that to your liking, but this is just the default that it will pre-calculate, which makes sense on average, right? There was a question? No. Okay. And now our model is trained and the thresholds are optimized, and we can go to the prediction. And for that, we're now going to use stacks that we haven't used during training, which includes both the validation and the training images. So instead of data/train folder, we're now going to use images from the test folder, which you haven't used so far. Okay, so this is important. We're just going to load those images, and then we predict the instances. And predicting instances from stacks means we first have to normalize them. So xt and yt is just for comparison, and xt are the test images that are still in the normal pixel counts. So we have to normalize them first with the same percentiles that we used during training. And then there's the function called model.predict instances. And this function just takes in as an input the image that you want to predict on, and it spits back a label mask and some details, which is a dictionary with polygon coordinates, with class probabilities if you have activated that branch, with a lot of more details per instance. And but the most important one is probably the label mask, and it will spit out some details. So here, for instance, it will get all the candidates. So it found 480 potential candidates. It will do this filtering, this non-maximum suppression, and it will filter out 51 of those 479 candidates will survive. And the resulting label mask will be stored in the labels. And here I then just plot both the ground truth and the prediction. And yes, if you see, they are all those hard cases that are not star convex. So in generally, it finds most of the nuclei, right? And if you see, like these labels were annotated in a way that it contained a lot of hard edges. And you see those hard edges are hard to reconstruct in 96 ways. So those hard edges will be actually star convexified, right? So this is kind of an effect that you see if you have crazy shapes, it will predict the most star convex shape according to that. So if you want to get that, you probably would have to use either a multi-class attachment and have a foreground background and use that as the center point of your watershed, right? Which is relatively easy to do, but just not part of the pipeline yet. But most of that is, yeah, it's quite nicely segmented.
But if there are cells which are not convex, some of them, it will not work at all, or what will be?
So it will essentially, so it depends on how non-convex are your cells. If they have, like, for instance, if you have neurons labeled that are filamentous, then it will just predict probably a lot of, like in those spaces where the neuron is a little bit thicker, it will predict something inside of that. Because the neurons, like filaments, are not star convex at all, right? If it's just cells with protrusions or something, it will predict the interior of the cell, right? And just miss the protrusion, right? So it kind of depends on what are your objects.
But can it cause a very long time for prediction? Because I tried my model and I figured out that there are some like difficult cells. And for prediction, even with GPU, it took me a very long time and I didn't get results in a understandable time. Can it be the problem?
Can you show me some of those images? Or?
Yeah, I can show.
Because it could be that that's actually an interesting point that has nothing to do really with.
There are some problems with like.
I think this sometimes can be problems with the NMS if you have too many candidates. If you have like 1 million candidates per tiny crop, then 1 million neighboring intersection tests will be done, or actually not 1 million, but 1 million log 1 million. So that can be quite the bottleneck sometimes.
I can share the screen briefly if I can.
I think you should be able now.
So here. I suppose it is like a very.
Okay. Yeah, but these things are probably, so it should just paint essentially the convex part interior of that. So that should work. So what do you see? You just see long prediction times.
Yeah, I usually like trying to run it in our cluster, and usually it's super slow. I can download everything, and after that, in predict instances, I have one this morning, and after that there is nothing. Even in error, there is no error, but I was waiting for a few hours, but.
So maybe that's something we can, we can go in a breakout room and you can prepare the error if that's possible. We can have a look quickly.
Thank you.
Afterwards. Sure.
Martin?
Any other questions? Yes.
Yes, you were showing in your presentation earlier on these orange cells, which are extremely non-convex, right? Have you tried to pretend it is, with a lot of overlap, then you're still predicting convex things, and then you're fusing objects which are touching?
Yeah.
The bottom right would be all separate. You might then, in a second step, fuse them by allowing a lot of NMS and say there's a lot of stars in there. It's a collection of convex shapes.
Yes, that is actually, that's a very, very nice idea. And that actually should work in principle. The only problem is that it's kind of fickly to understand when do things are separate objects that just happen to be close and when they actually are belong together. So that is the main core, right? And if you just want to stitch things together, it's not clear how to get the information that they should not be actually two objects, but one. So it should be fused and one not. And the hardest problem is to make it in such a way that it's robust, not only for a simple use case that I have, but to turn it into something that works for almost every problem that people have. So that is making it really robust. This is something that you can try maybe if you have a pipeline and then you extend the library in some ways to make that for your own use case, and then it would work. But I haven't thought of, or we haven't come up with a way to make it in such a way that it will work robustly across different data sets, because a lot of thresholds.
Yeah, could you use superpixel? Could you use superpixel type of thing where you network, and then you're based on certain properties of the network? If there are then certain connectivity, you would count it.
You could actually, you could make like every individual instance prediction. You could turn that into a supervoxel and just have edges between overlapping things, and then define a criterion. When is this? What is the cost of that edge? And then do this fusing aggregation based on that superpixel graph. That is true, yeah. But it's something that is, it's something that has to be tuned per problem, and it's not something that is, but it's a good, it's a good idea. Any other question? Everybody went through the thing and could run it, or?
I have a question.
Yes.
So this pipeline, will it automatically, if we have multiple channels, then will it just work on all the channels at
Yes, it will just use all the channels. Mm-hmm.
Okay. And
so I think you have to make sure that the channel is like, if you save it as TIFF, then the channel should be, I think, the first or the last. Yeah, the channel should be the last dimension of the array if you load it in Python, but you can always reshape it, of course, in Fiji or wherever you want to. It will complain if it's not so.
Okay, and then I can have multiple channels.
Then it will use.
So there will be just at the end of the as the last dimensions, there will be the channels.
Exactly, and it will use all the channels that it sees by definition.
Mm-hmm. Mm-hmm. And can we somehow tune it that if we know that certain certain things are in certain channels, like?
That's a good question. Yeah, you can do it now, but you would have to extend the network. So what that's an interesting question. Yeah, what you would do is then, but that is research, right? That is not something that can be tried and tested. But of course, it's super interesting if you have like 3 channels and 2 of them are important and the other one is super non-important for your decision. You could imagine having kind of a channel-wise attention where you multiply kind of a weight for each channel, which tells you, is this important or not? And put that as the first layer and make these weights trainable, right? And you can do that now with keras. You can kind of stack models on top of each other. So you could use that layer as one first layer and then stick on top of that a stardust model. That should work. But it's a bit, you have to dig into the code, and that's a bit, but it's super interesting.
Or I was even more thinking that if I have multiple multiple labels of objects, and then I know that for a particular label, it should look in this channel, or like I have a type of cell, and then that cells will be marked with green channel, and then another type of cells will be just marked with yellow channel.
So if you just want to, if you want to segment all of those cells, it will just learn which channel is relevant for which. So this.
Okay. So
I was just thinking more if you want to exclude. Sometimes you have a phenotype, and you don't want that to be relevant for the segmentation, right? You want to exclude that channel because you want to do base measurements based on that channel, and it should not be used really for the actual measurement. Then you should not include it into your channels because it will use that if it can, right? It will use whatever it gets.
Okay, so, I understand. Yeah. Thank you.
Just to clarify on that question as well. So does that mean across the channels you have, like a single channel doesn't necessarily need to have all the information for segmenting an object, as long as across all the channels it's able
exactly.
to
So it could be, for instance, sometimes you have, like, imagine you have two phenotypes, two expressions of a nuclear marker that depends on the cell state or on the cell morphology. Phenotype is expressed in one channel and not in the other, and in the other channel it's in this channel and not in the other. But together they would populate, or they would consume all the different outlines that you want to get. Then you could just paste those two channels as an input, and it will learn that it should use essentially both of them simultaneously, right?
Yeah.
Or sometimes you have a nuclear marker, and then you have for some of them it's not expressing, but you have a membrane marker that is active for these instances, and it will just use both of them. If this is what you want, right? If you don't want this specific marker to be relevant for the decision, then just exclude it from the training.
Yeah. So if we do like cell segmentation, could you just segment the whole cell like without doing the nuclei?
Yeah, sure. But then the problem is always to find the correct C. So if you have a lot of touching cells, to find correct instances, but then you would have to kind of annotate the nuclear region that is star convex, use that, predict that, and then do some expansion step based on a probability map that you could get out or other means. So you would have to do an additional step. Yeah.
Okay.
Any more questions? Anybody already tried to annotate his own data? Or her? Everybody exhausted? I can hear you, Mark. Your mic, yeah. Sorry?
Good to try it.
Yeah. I mean, we can, what we can do is we can just do whatever your current use case is. We can just, you share your screen and we can give advice what would work best if you have questions, because that's what we're here for.
You mentioned earlier that it's better to not have in Fiji the Z as a Z, but to use it as a time series. Notice in Python, it's always expecting a time frame. It will complain if you give it as a Z instead of a T. Is there a reason for that?
No, this is something super specific for LabKit, because LabKit uses Big Data Viewer, and Big Data Viewer, like if you rotate if you place your mouse and rotate a little bit, you suddenly are rotating along Z, and it's hard to get back. So it's a bit hard to step through each Z plane. And in Big Data Viewer, you can step through time points easily, right? Without missing a time point. That's why if you map that before, you can precisely go to every Z plane of your liking, right? And not miss someone in the middle. That's something, because you cannot do it stepwise. It's a purely pragmatic choice for this tool. Any other questions? Or
hi.
Hello? Yes, there's some.
Hi, Martin. It was a great talk, and for sure I will try it on my data. But I have a question. I have this annotation already with the QPath. So how do I use the StarDist on the QPath annotation?
So you have annotations and want to train your own classifier or your own model.
Wait, I can't hear you.
I can't hear you.
So something with it.
Yeah, I thought I was the one providing feedback.
Yeah, now I can hear you. Sorry.
Ah, okay. Now I was very confused. Oh yeah, no, yeah. So you have your own. So you have already made annotations in QPath and want to train a model based on those annotations. Is this correct?
Yeah.
Okay. So the easiest way is actually if I share my screen. So if you have already annotations in QPath, we made some scripts that you can use within QPath, and these are called QPath export annotation, Groovy scripts. They're actually in the main repo. If you go to StarDist main repo under extras, and I can put the link into chat. And this script, if you just put it into QPath, it will automatically extract all the rows that you have and save it as a format that StarDist can understand. Okay, so this, I'll just put it into chat. And this is, you can use and then use that in the Python notebooks to train your own models. That makes sense.
Okay, I will try. Thank you.
Any other questions?
Yes, can I ask about hyperparameters for the network? If I want to train my own network, use in your code, the configuration file, there are train patch size, and you said that adjust for your data, make patch size as large as possible. Do you have some advices how to find a trade-off between memory issues, so not to go out of memory, and what is the patch size? It depends on the data we have. For example, in my case, I have 100 by 100 pixels, approximately one object.
So then your patch size should be probably 196-ish along this dimension. And in Z, it's less, right? Or
yeah, in Z, it's like 5 times, how to say, Z scale and 5 times.
So I would always at least use like twice the size of the object along each dimension. And if that doesn't fit into memory, then just set the batch size to 1, because then just one single batch is used, and then just make it as large as possible, essentially. Right?
Yeah, I already did by batch size 1, but yeah, okay.
Because if it doesn't fit, then you have to use the grid parameter. I would actually, for your case, just use grid 4. So.
Oh, maybe it would be better. Yeah, because now
because you have very large objects, and then it doesn't matter.
So small patch size doesn't work for my data, but yeah, grid size would be a good solution. Thank you.
Sure. Actually, I restarted the training with a little bit more epochs, and I think, so the epoch loss TensorBoard is strange. So you see it trained a little bit more, and it's still, I mean, it keeps on going down and down. It will be here at some point and will be twice as good, but it's already okay. And if you look at the intermediate output, it starts to look a little bit better, right? I'm gonna reload it. So this is the object probability, right? We predict those two things, object probability and distances, and this is the object probability. You see at the beginning, it actually predicts the holes, and it gets better and better, and suddenly realizes what are the centers of objects, right? And same for the distances at the beginning, and this is just 3 distances color coded, so 3 random or 3 from the middle. So I think the 10th, the 20th, and the 30th. So this should resemble edges, and if you come now, suddenly it starts to get into this distance map shapes, where things are bright at one side and things are dark at the other side. And it's very nice to look at if that number is not 21 but 200, then you can really see how it snacks into